how we can predict the structure, why we need to predict the structure actually, like why we need to predict the structure. We will be covering all these aspects of protein structure prediction. And first of all, you already know better even than me, like what is protein, what is amino acids. So start with, we have, if we want to go through the structure, we have different levels of protein structure. And the first one is the primary structure. And the primary structure is actually the sequence, the amino acid sequence, yeah, or amino acid residue sequence. So when we have a sequence of these residues, we will be calling that as a primary structure. And if this primary structure is locally folded, it gives rise to a secondary structure. So local foldings occurs in the primary structure, and that gives rise to the secondary structure. And once a whole chain of a protein, they got some conformation, some 3D conformation, that will be tertiary structure. The global conformation means end-to-end conformation of the whole chain, that will be giving rise to a tertiary structure. And quaternary structure means when there is assembly of 2 or 3 or more tertiary structures, they combine together, or they will mean they assemble together, they group together to form a complex structure, and that assembled structure will be a quaternary structure. Means you can see here there are 4 or 3 or 2 different types of structure, like 2 different colors, they are represented with 2 different colors. So that is the combination of 2 different chains. So the combination of different chains will give rise to a quaternary structure. So sequence comparison so this is a bit different aspect from this one, and that is like how we can determine a conserved region within a sequence. So we need to align multiple sequences. So multiple sequence comparison of multiple homologs of particular protein revealed highly conserved regions that are important for function. So what we actually do, if I want to know the conserved region or an important region within this protein sequence, what I will do, I will align similar sequences of this sequence, like I will align different isolates of this sequence. And once I align them, I would know there are certain differences within this alignment. There will be certain differences, like that they align here. There will be some differences. There will be some differences here, and there will be some conserved regions. And if I mean, this is like conserved region here. So this region is conserved. So the conservation of this region means this region is important for certain specific function. And the nature has kept this region conserved and changed the remaining sequence. So this region is now the conserved region. And how I determine this region? By aligning multiple sequences of the same homologs. Means they are same homologs. So I align them, and by aligning these sequences, I got to know the conserved regions within these sequences. And next to it is clusters of conserved residues are called motifs. So these are conserved residues. They cluster together, and they will be called as motifs, and they carry particular function, are form of a particular structure. So that is important for conserved protein. Means within this protein, there will be conserved region, and that conserved region, or simply that motif, is responsible for some important function. And if you can see here in this alignment, their different sequences has been aligned together, and this region, the boxed region, is called, they call it a motif because that is conserved. And if you can see this gray color, that will mean this gray color where is a small hydrophobic region, small hydrophobic amino acid. And the other one, the dark shade, that will be large hydrophobic amino acid. And the green one, they will be polar amino acid. So if you can see, the polar are being replaced with the hydrophobic, the larger hydrophobics here in this very specific case. Here, some positive charged amino acid, like you can see this one, this residue, they are charged, positive charged amino acids. And this negative charged amino acid, these are negative charged amino acids. So we can say within this motif, there are certain residues, they will be conserved throughout. Like here you can see this is the same the residue with having same properties, and here you can see. So this motif will be conserved throughout the sequence, and this conservation means there is specific function related to this motif. And if you can see here, the green one, uh, they are invariant, means the regions, the residues that always remain the same, like the same amino acid, aspargine. And the conserved, the yellow one are conserved. The residue is generally similar. For instance, negatively charged amino acid means the yellow region are the amino acid, even though they are different amino acids, but they will be negatively charged. Means if there is a sequence and that is negatively charged. If we have here some example of that, no, we don't have it here. Okay, so and the gray one, they are non-conserved. They can be of many different residues in different species. Am I clear up till now?
Yes, sir.
Okay, who will explain?
So मैं करूँ?
जी please.
एक्सक्यूज मी सर,
जी
ये
जी,
जो
कोई-
इसके अंदर आपने मोटिव को एक्सप्लेन किया है, इसमें कुछ रीजन्स मैच नहीं कर रहे,
जी इसमें कुछ
उनका
रीजन,
क्या?
जी उनमें इसमें असल में इस तरह रीजन देखे जाते हैं जिसमें मैक्सिमम रीजन जो हैं, मैक्सिमम जो रेजिड्यूज हैं वो मैच कर जाएं। तो यहाँ पे मैच बहुत ज्यादा हो रहा है, यहाँ पे मैच हो रहा है, तो इस वजह से इसको फिर कंसर्व कहा जाता है। और नॉर्मली इसको रिप्रेजेंट इस तरह किया जाता है कि दिस इज कंसर्व, दिस रीजन इज कंसर्व, ये कंसर्व्ड है। यहाँ पे फिर इसको एन लिखा जाता है, ये एक्स लिखा जाता है कि कोई भी रेजिड्यू यहाँ पे आ जाए, लाइक यहाँ पे।
ओके सर, कंजर्व के लिए फिर 100% कंजर्व्ड इम्पोर्टेंट नहीं है?
नहीं, एग्जैक्टली 100% कंसर्व्ड
Okay, sir.
इम्पोर्टेंट नहीं है, बिल्कुल।
Okay.
जी, हु विल एक्सप्लेन?
सर आपने यहाँ पर अभी तक ये बताया कि सबसे पहले जो है वो प्रोटीन के 4 डिफरेंट स्ट्रक्चर्स होते हैं। जैसे कि सबसे जो सिंपल स्ट्रक्चर होता है वो प्राइमरी स्ट्रक्चर होता है, उसके अंदर सिंगल चेन्स होती हैं अमीनो एसिड्स की, और अगर वो थोड़ी सी मॉडिफाई हो जाए, वो हेलिक्स की फॉर्म बना ले, तो उनको हम कहते हैं सेकेंडरी स्ट्रक्चर्स। और अगर ये सेकेंडरी स्ट्रक्चर्स जो है वो आपस में कोई कंफर्मेशन जो है वो अडॉप्ट कर लें, उसको हम कहते हैं टर्शियरी स्ट्रक्चर्स। और दो या तीन टर्शियरी स्ट्रक्चर जब आपस में कंबाइन करते हैं, लिंक करते हैं, तो उनको हम कहते हैं क्वार्टरनरी स्ट्रक्चर्स। उसके बाद जो है वो हम लोग ये बात करते हैं कि अगर हम लोगों ने किसी भी प्रोटीन के स्ट्रक्चर को, उसके अंदर कंजर्व सी रीजन्स को प्रिडिक्ट करना हो, तो उसके अंदर हम सबसे पहले क्या करते हैं, हम उनके मल्टीपल सीक्वेंसेस को अलाइन करते हैं और उसके अंदर देखते हैं कि सबसे ज्यादा कंजर्व सीक्वेंस सबके अंदर सेम कौन से हैं। और जो कंजर्व सीक्वेंस होते हैं वो समझे जाते हैं कि ये इसके प्रोटीन सर्टेन फंक्शन के लिए बहुत इंपॉर्टेंट है। उसके बाद मोटिव्स के बारे में बताया कि मोटिव्स के अंदर, प्रोटीन के अंदर जितने भी कंजर्व रेजिड्यूज होते हैं, अगर उनका जो क्लस्टर्स होता है उनको हम मोटिव्स कहते हैं। और इनका काम क्या होता है कि इनका एक सर्टेन फंक्शन होता है प्रोटीन के अंदर, या तो ये कोई कंजर्व रीजन बना रहे होते हैं प्रोटीन के अंदर, या इनका कोई सर्टेन फंक्शन होता है। जैसे इनके अंदर कोई स्मॉल हाइड्रोफोबिक अमीनो एसिड्स आ सकते हैं, लार्ज हाइड्रोफोबिक अमीनो एसिड आ सकते हैं एक्सेट्रा। इसके अलावा 3 टाइप्स और हैं, जैसे इनवेरिएंट अमीनो एसिड्स होते हैं। इनवेरिएंट वो रेसिड्यूज होते हैं जो हर प्रोटीन के अंदर सेम रहेंगे, लाइक एस्पार्जीन। और कंजर्व सीक्वेंसेस वो होते हैं जो अमीनो एसिड डिफरेंट हो सकते हैं लेकिन वो जनरली सिमिलर होते हैं, जैसे नेगेटिवली चार्ज्ड अमीनो एसिड। अमीनो एसिड डिफरेंट हो सकते हैं लेकिन उनका जो कैरेक्टर है वो नेगेटिवली चार्ज होगा। और कुछ अमीनो एसिड ऐसे होते हैं जो कंजर्व नहीं होते हैं, वो डिफरेंट रेसिड्यूज होते हैं, डिफरेंट स्पीशीज के अंदर जो है वो डिफरेंटली बिहेव करेंगे। तो आपने अभी तक ये बताया।
ठीक है, well done. But I want to correct myself for one thing. This is aspartic acid, ASP. Aspargine will be ASN.
ओके सर।
Okay. And next to it is the protein architecture. So normally, as we know, the amino acid, they are linked by a peptide bond. Like these are two amino acid, like given here. There are two amino acids. This is one amino acid, and this one is the other amino acid. They are linked together, and if they are linked together by releasing one water molecule, and they will be linked together, and this will be a peptide bond between the carboxyl group and the amino group. And later to it is okay. So amino acids are actually linked together by a peptide bond. This bond will be called as a peptide bond. And each amino acid, as you know, each amino acid will be having a central carbon. This will be a central carbon, you can see, and amino group, a carboxyl group, a side chain, and this carbon will be called as chiral carbon because it will be having four different molecules attached to it. Like it will be having four different molecules. So this will be called as a chiral carbon. So amino acid normally have this chiral carbon, and this is actually the central property of an amino acid, except is for glycine. And okay, so this carbon, so we have the bottom line is an amino acid will be having a chiral carbon with a carboxyl group, amino group, alkyl group, and a hydrogen ion, hydrogen. And next to it is, so this is a general amino acid structure. You can see this one. This can also be represented like this one. And here, that is alpha amino group. That is this one is alkyl group, the side chain. This one will be alpha carboxyl group, and this is the center. This one is which we called previously as chiral carbon. So this chiral carbon is actually alpha carbon because this will be alpha, beta, and the nomenclature actually goes on if we have further if we have some classification based on this that how we name these amino acids. So this is regarding that. So this one will be alpha carbon, and this is actually the chiral carbon. So after that, there are how the amino acid, if this is the basic structure of an amino acid, how that differ from one another. The difference between these amino acids, they occur in this alkyl group, this R group, and certain modifications occur in these amino acids. And the first of the modification is phosphorylation. Phosphorylation normally occur in the alkyl group if that is added with if that is hydroxyl group here, and that hydroxyl group is replaced with the phosphorylation, means phosphate group. And normally this phosphorylation occurs in which residues? In serine, threonine, and tyrosine. These three are the golden examples of phosphorylation. So these three residues are they are commonly phosphorylated in different proteins: serine, threonine, and tyrosine. So they are the candidates for phosphorylation. And next is N-glycosylation. N-glycosylation is when glycosylation are the addition of sugar that occurs at the amino group site. Amino group that is with the alkyl group, and amino group when that is modified and addition of sugar occur at the amino group of the alkyl group. So that will be N-glycosylation, and aspargine is the golden example for that. O-glycosylation is when sugar is added with the hydroxyl group with the O, and that will be the O-glycosylation normally occurs in serine, threonine, and some modified residues. Hydroxylation that occurs in proline and lysine. Similarly, glycosylation that occurs in glutamine. This carboxyl group that has been added to the alkyl group of this amino acid. So carboxylation normally occurs in glutamine. Similarly, disulfide bond formation also occur, and that is when two cysteine they come closer together and making cysteine. So oxidation actually occurs here, and that will be actually the removal of hydrogen. Am I clear up till now?
Yes, sir.
Yes.
Okay, who will explain?
Sir,
Ji, Kinza, please.
सर इसमें बेसिक प्रोटीन आर्किटेक्चर बताया है कि अमीनो एसिड लिंक होते हैं बाय पेप्टाइड बॉन्ड होती है CNH और O की डबल बॉन्डिंग पे। उसके अलावा ईच अमीनो एसिड होता है वो उस पर एक सेंट्रल कार्बन होता है, अमीनो ग्रुप होता है, कार्बोक्सिल ग्रुप होता है और एक साइड चेन होती है। अब जो लेफ्ट साइड पे सबसे नीचे वो एक जनरल अमीनो एसिड स्ट्रक्चर है। उसके अलावा जो केमिकल एनाटॉमी ऑफ अमीनो एसिड है उसमें हमारे पास एक आर ग्रुप होता है जो साइड चेन होती है। अल्फा अमीनो ग्रुप होता है, अल्फा कार्बन होता है जिसको कार्बन कार्बोक्सिल ग्रुप होता है। अब डिफरेंस इन साइड चेन डिस्टिंग्विश एसिड से के
आपकी आवाज
मॉडिफिकेशन होती है जिसमें डिफरेंट फॉस्फोराइलेशन होती है सर सही आ रही है? हेलो हेलो सर।
जी जी अभी ठीक है आपकी आवाज, बताएं। अच्छा, who will explain next? जी
मैं कर दूं?
शफीक प्लीज।
सर इसमें जो प्रोटीन का स्ट्रक्चर है, बेसिक स्ट्रक्चर, उसके बारे में बताया गया कि जो हमें पता है कि प्रोटीन जो अमाइनो एसिड रेसिड्यू पे कंसिस्ट होती है, तो ईच अमाइनो एसिड एक दूसरे से पेप्टाइड बॉन्ड के थ्रू लिंक हुआ होता है। और पेप्टाइड बॉन्ड होता है एक अमाइनो एसिड का अमाइन ग्रुप और दूसरे अमीनो एसिड के कार्बोक्सिल ग्रुप के दरमियान जो वाटर की रिमूवल होता है, उसके बाद पेप्टाइड बॉन्ड बनता है। इसके बाद हमने बेसिक स्ट्रक्चर देखा कि एक अमीनो एसिड किन किन चीजों के ऊपर कंसिस्ट होता है। एक उसके अंदर सेंटर कार्बन एटम का होना लाजमी, जो कायरल कार्बन होता है, जिसमें उसने 4 साइड उसकी ऑक्यूपाइड होती है और डिफरेंट फंक्शन ग्रुप उसके साथ अटैच होते हैं। उसके अंदर एक कार्बोक्सिल ग्रुप अटैच होता है, उसके साथ एक अमाइनो ग्रुप अटैच होता है, एंड साइड चेन यानी अल्काइल ग्रुप कोई भी साइड चेन आके अटैच हो सकती है। उसके बाद एनाटॉमी में हमने देखा कि जो कायरल कार्बन के साथ अमाइन ग्रुप अटैच होगा उसको हम अल्फा अमाइनो ग्रुप कहेंगे, और सेम इस तरह कार्बोक्सिल ग्रुप और सेम इस तरह अल्काइल ग्रुप के साथ। और बाकी अगर कोई मॉडिफिकेशन होती है जैसे लाइक ग्लाइकोसाइलेशन यानी ग्लूकोज ऐड होता है, या हाइड्रोक्सिलेशन होती है, या फॉस्फेट ग्रुप अटैच होता है, तो वो किन किन रेसिड्यू के ऊपर अटैच होगा वो हमने देखा था साइड।
ठीक हो गया, ठीक है, गुड। एंड नेक्स्ट टू इट इज so the alpha helix क्या है? Alpha helix, so these are actually the secondary structures now we are talking about. So we are done with the primary structure. Now we are concerned about the secondary structure. So the alpha helix, the carbonyl oxygen of the residue I. So that will mean the first residue in a sequence forms a hydrogen bond with the amide of the residue I plus 4, means the fifth one, the fifth amino acid. So the first and the fifth amino acid, they will form hydrogen bond. So keep in mind hydrogen bond. So this is a hydrogen bond. So due to the hydrogen bonding, the overall conformation that they got a structure, and that structure will be called as alpha helix. And what alpha helix means, the I residue with I plus 4 residue, they form hydrogen bond together and although each hydrogen bond is relatively weak in isolation, so if we consider one hydrogen bond, that will be weak, but the sum of all hydrogen bonds that in a complex in the whole conformation that they give rise to a quite stable conformation. And the propensity of peptide for forming an alpha helix also depends on its sequence. So that is the sequence which determine what conformation it will make. Like it will make alpha helix, it will make beta sheets, it will make turn. So that will be depending on the sequence, actually, on the primary sequence. And what actually beta sheets are. In a beta sheets, carbonyl oxygens and amides form hydrogen bonds. If you can see that the hydrogen bonds, that they are making hydrogen bonds, and these secondary structures can either be anti-parallel. So they can be either be anti-parallel, like here. These two sheets are anti-parallel, or they could be parallel. So and need not to be planar. They cannot be planar. They can be twisted as well. So in some cases they could be parallel. In other cases they could be twisted. And the propensity of a peptide for forming beta sheets also depends on the sequence, on the primary sequence. And next to it is a beta turn. A beta turn is defined by 4 consecutive residues like I plus 1, I plus 2, and I plus 3 that do not form alpha helix and have a C alpha I and C alpha I plus 3. So the bond will occur between I residue and I plus 3 residue, and the distance should be less than 7 angstrom. And the turn lead to a reversal of the protein. So here you can see the protein reverts back on itself. And beta turn all allow the protein backbone to form to make abrupt turns. So therefore that is called a beta turn. And again, the propensity of a peptide for forming beta turns depends on its sequence. Am I clear up to now?
सर ये प्रोपेंसिटी को थोड़ा एक्सप्लेन कर दें प्रोपेंसिटी
So
वर्ड को
it means how we would know a sequence will be turning into a beta turn, or it will make like how we would know like a sequence will make beta turn, beta sheets, or alpha helix. How would we know, or what or on what things this conformation depends? So it says the secondary structure formation depends on the primary structure. So if we have a sequence, we will be having an assignment for that as well. Like if we have a sequence like GT, like G M G X, if this is a sequence and something, so how we could either we could determine this sequence will make a beta sheet or alpha helix or a beta turn. We can determine this thing by determining the sequence. Simply from this sequence we can determine either this sequence will make a beta sheet or a beta turn or a alpha helix. We can know that from the sequence, because this sequence will give rise to the secondary structure. Am I clear or not?
यस सर
So GG.
सर अल्फा हेलिक्स और बीटा टर्न में एक दफा मेन डिफरेंस बता दें.
देखें अल्फा हेलिक्स में क्या था, that is, if you can see here, that is the bonding between I and I plus 4. So I plus 4 is the 5th residue, and here the bonding between I and I plus 3, that one is the 4th residue. So that will means if the bonding between I and I plus 4, that will be this interaction is more farther, and here in beta turn it will be nearer, like it will be like this, the 3rd residue. So it will be the 1st and the 5th residue, and this will be the interaction between the 1st and the 4th residue. So that will abruptly turn the sequence actually.
सर ये हमेशा फोर्थ रेजिड्यू ही होगा बीटा टर्न के अंदर.
Exactly.
ओके सर.
So who will explain? जी Fatma Ramzan, please.
Yes, sir.
जी.
सर इसमें आपने अल्फा हेलिक्स का बताया है. 3 फॉर्म्स ऑफ सेकेंडरी स्ट्रक्चर्स होते हैं, अल्फा हेलिक्स, बीटा शीट्स और बीटा टर्न्स. अल्फा हेलिक्स में ये है कि कार्बोक्सिल जो ऑक्सीजन है वो फर्स्ट और फिफ्थ के बीच में हाइड्रोजन बॉन्डिंग करेगी और उसका डिस्टेंस थोड़ा सा दूर होगा और इस तरह फिर वो अल्फा हेलिक्स बना सकते हैं. और जो हाइड्रोजन बॉन्ड है वो वीक होते हैं आइसोलेशन में, इसलिए जो उनका जो सम होता है हाइड्रोजन बॉन्ड का वो बैलेंस करता है एक दूसरे को. और फिर आपने बताया है कि बीटा शीट हैं. बीटा शीट में ये है कि कार्बोक्सिल, ऑक्सीजन और अमाइड जो हैं वो हाइड्रोजन बॉन्ड्स बनाते हैं और ये 2 तरह की कंफर्मेशन हो सकती है, पैरेलल हो सकती है, एंटी पैरेलल हो सकती है. फिर बीटा टर्न्स है जिसमें 4 कंसेक्टिव रेजिड्यूज होते हैं 1 से I से लेके I3 तक, जो एक दूसरे के साथ बॉन्डिंग बनाते हैं और वो कंटिन्यूस होते हैं, इसीलिए वो अल्फा हेलिक्स नहीं बनाते मगर वो फिर बीटा टर्न बनाते क्योंकि वो एब्रप्ट टर्न्स होते हैं, एकदम से उसको चेंज कर सकते हैं वो. और फिर आपने बताया कि प्रोपेंसिटी होती है, जो एक एबिलिटी होती है स्ट्रक्चर की कि वो बीटा टर्न बनाएगा या बीटा शीट बनाएगा और या अल्फा हेलिक्स, वो उसके प्राइमरी सीक्वेंस के ऊपर डिपेंड करता है कि वो किस तरह का.
Exactly. Okay, so
सर प्रोपेंसिटी जो है वो तो सीक्वेंस के अपने ऊपर डिपेंड नहीं कर रही होती. लाइक हर सीक्वेंस का खुद से देखना है कि इसमें कितनी एबिलिटी है कि कौन सा स्ट्रक्चर बनाएगा.
Exactly. So this is simple. Like if that is a sequence and that sequence will be like this. So it will be having, if that is I and I plus 4 residue interaction. So इसने इस तरह का स्ट्रक्चर बना देना है. अगर और ये क्या किस पे डिपेंड कर रहा है, इस रेजिड्यू पे डिपेंड कर रहा है और इस रेजिड्यू पे डिपेंड कर रहा है क्योंकि ये आपस में इंटरेक्ट कर रहे हैं. ठीक है. So they are interacting with one another, making like this structure. And if that is like a sequence is like this, so they are interacting like that is I and I plus 3. So that will mean the abrupt turn, that will be abrupt turn, and this will be a like loose structure like that. Okay. And if that is sheet, so that will mean if it will be like this one. So if this is anti-parallel, some could be anti-parallel. In some cases, that could be parallel. They can be parallel. Okay. So okay. So and they are like they can, so that is not like I plus 3 or I plus 4. That is in किसी भी रेजिड्यूज के दरमियान हो सकते. सो दैट डिपेंड्स ऑन द रेजिड्यूज. ठीक है. Am I clear?
Yes, sir.
And next to it is the conformation flexibility. So a backbone conformation, torsion or rotation angle bond angles around. So how much this sequence is either this, if this is an amino acid sequence, so how much is this sequence flexible, how much they can move, how much the sequence can move. So in our mind, normally we think this is a rigid structure, but it is not like so. It has flexibility, it can move because this whole structure can move. Like this one, this whole structure can move, this whole structure can move, and similarly this one can move. So they are different planes, and that these different planes can move together. So this can move, so this can move differently from with in context with this plane, and this can move in context with this one. So they can move in the context of one another. And this movement is normally the torsion or rotation angles around this carbon nitrogen bond, this bond. So we can see, if we can see this carbon alpha and nitrogen bond. So this bond is known as Phi bond. This Phi bond between carbon alpha means that chiral carbon and this nitrogen. This is the alkyl group, actually, this one. So the carbon and the alpha carbon and the nitrogen will be called as this bond, or the moment of this bond, this angle. So this bond in context with this plane, so this will be called as alpha bond or alpha angle. So this will be alpha bond. Similarly, this chiral carbon and this carbonyl carbon, so this bond will be called as Psi bond. So this is Phi bond between carbon nitrogen, and between carbon carbon, the chiral carbon and the alpha carbon and the carbonyl carbon, this will be called Psi bond. So two bonds are important basically. The first one is Phi bond, the second one is Psi bond. So there are two major bonds between them, and one is omega. We will be talking about that later. But here we are been discussing the Phi bond and the Psi bond. And these Phi and Psi give flexible conformation to the whole protein. And because of these bond flexibility, this whole protein can move and can attain different structures. And this and one more important thing, it will not move like freely. It will have a certain limitation, and that limitation is sterical hindrance. And there are sterical hindrance occurs in proline, like the properties that normal amino acids will have. Two of the amino acids will have different properties from the remaining 18 amino acids. And the first one is proline. Proline will be having sterical hindrance, or it will be having different properties than the remaining of the amino acid, and that is because the amino group is being bound with the alkyl group. It will make hydrogen bonding with the, if you can see, this is the amino group of the amino acid that is bound together with the alkyl group. So it will not be like the normal amino acid. The proline is not like normal. Its properties will be different from the normal amino acid, because normal amino acid will be having a chiral carbon with the carboxyl group here, and here that is a carboxyl group, and here that will be a hydrogen, here that is alkyl group, and here that is amino group. But in this case, this amino acid group is bound together with this alkyl group, and therefore proline is normally different from other amino acid. Similarly, glycine is also different from other amino acid, because in place of alkyl group there is a hydrogen bond. And if you can see, the hydrogen bond is there and there is no alkyl group. So this is no more chiral carbon, because it will be having two same groups within the four, and therefore these two amino acids, they will behave differently from the remaining amino acids. Am I clear?
Yes, sir.
And here within the sequence, amino acid sequence, normally we write from the amino terminal to the carboxyl terminal. Here we can see this is the amino terminus, and this will be the carboxyl terminus. Who will explain? जी please.
सर मैं करूं?
खंसा कर दें, इसके बाद हिजाब, नेक्स्ट स्लाइड आप हिजाब में करना.
सर आप
सर करूँ एक्सप्लेन?
जी प्लीज।
सर आपने इसमें बताया कंफर्मेशनल फ्लेक्सिबिलिटी का कि ये जो बाउंड होता है ये रिजिड नहीं होता है, ये मूव करते हैं, रोटेट करते हैं किसी भी एंगल पे। जब मतलब कार्बन और नाइट्रोजन के दरमियान बाउंड हो, जिसे हम अल्फा बाउंड कहते हैं, उस पे, और साई बाउंड जो कार्बन और कार्बन के दरमियान होता है, ये स्टेरिकल हिंड्रेंस शो करते हैं। सिवाय दो अमाइनो एसिड जैसे प्रोलीन और ग्लाइसिन, इनका नॉर्मल अमाइनो एसिड से स्ट्रक्चर थोड़ा डिफरेंट होता है, इसलिए इनकी मूवमेंट और अमाइनो एसिड की मूवमेंट का फर्क होता है। हमारे पास अमाइनो एसिड का सेम टर्मिनल जो होता है, अमाइनो एक तरफ अमाइनो होता है, एक तरफ कार्बोक्सिलिक होता है। और सर आपने इसमें फिर वो स्ट्रक्चर को एक्सप्लेन किया, प्रोलीन के स्ट्रक्चर को, कि यहाँ पे जो है वो नाइट्रोजन और हाइड्रोजन के साथ बाउंड होता है, जबकि नॉर्मल में हमारे पास वो अमाइनो ग्रुप होता है, और नीचे आर होता है, मतलब, और ऊपर साथ पे कार्बोक्सिलिक ग्रुप होता है और उसके साथ बाउंड होता है। लेकिन जब प्रोलीन है, इसका स्ट्रक्चर थोड़ा डिफरेंट, सिमिलरली ग्लाइसिन का भी स्ट्रक्चर थोड़ा डिफरेंट है, तो ये रोटेशन शो करेंगे। इसलिए जो आई गेस सर आपने बताया था कि जो ज्यादा हिंड्रेंस शो करेगा वो स्टेबल है, गैस मोड जो लो करेगा शो।
नहीं, ये स्टेबल की बात नहीं हो रही। Here actually why we study them, because we want to know the structure of amino acid. हम amino acid जो protein sequence है उसका structure determine करना चाह रहे हैं, so we need to know the principle, we need to know the basic principles, like we need to know the rules of a protein. So if we have a sequence, we can determine the secondary structure, and by secondary structure we need to know other amino acids will not behave differently, but if we have two amino acids, we will be taking care of these two amino acids. If they have there in the sequence, they will behave differently from the other amino acids, and that's simple. आगे जाके हम we will be discussing things like how we can determine the secondary structure actually. Am I clear? Okay.
Yes, sir.
So that doesn't mean they will be more stable or less stable. We are just talking about how they will be behaving, or how flexible will be they will be flexible, and how they will behave in the secondary structure. And next to it is this Ramachandran plot. This is important, and the whole secondary structure depends on this plot. There was a biophysicist, and this biophysicist G.N. Ramachandran constructed a space filling model of peptides and made calculations to determine which phi and psi are sterically permitted in a polypeptide chain. So how we could know either this whole plane can move here, like if I could draw, if I would know, I would want to know either this whole plane, either this can move from here to here as well, like if this can move from here to here. So the protein conformation is now different. If it moves from here to here, from here to here, it will now different. But actually it doesn't occur like this. It can move up to certain limits. It doesn't move like in 360 angles, because by moving here, it will collapse with this. It will be, because it will be having a space. It will, this will be having, it will carry a space here. So it can not move like more, it cannot move freely in all directions. And this is called, this will be called as, this will be determined in space filling model, because every atom will be having, it will be carrying some space, and if that is filled space, that in that model we can determine that that's phi and psi is permitted to have that, and to have certain shapes because of these the permeability, because of the limits of these phi and psi bonds. And it also provides a simple weave of the conformation of the protein. So from this, we can, from these phi and psi angle, we can determine the conformation of a protein, the structure of a protein actually. So from this secondary structure can be determined by these phi and psi angle, from these phi and psi bonds. So simply, if we could know these bonds, the angles that these bonds could form, so we can determine the secondary structure. Is simple, simple. And if you can see from this, so this, from this plot, this one is known as, this plot is known as Ramachandran plot, also known as Ramachandran diagram, or also it is called Phi and Psi plot, originally derived in 1963 by this scientist. So here, if we can see the this chart or this plot, here you can see the phi. On the x-axis, this is the phi bond. On the x-axis, this is phi bonds, and this one is psi. So this is the phi angles, and this one is the psi angles. And from this, we have different positions. The different colors has been represented, and that is represented in different color. What this red region shows, this red region shows the amino acids that are allowed and that are in the allowed region, namely alpha helix and beta sheets conformation. So this will be the beta sheets. They will come here. So the amino acid making phi angle in this area and psi angle in this area, in this region, there will be beta sheets. Here in this region, there will be right-handed alpha helix. So that will be phi will be here and psi will be here. So they will be making alpha helix, right-handed alpha helix. And what the yellow region shows, the yellow region shows is the outer limit. So here the amino acids, if the amino acid is here, so this will be right-handed alpha helix, this one. And the what is the yellow region shows, the yellow region is the outer limit. And what actually the white region shows, the white region is sterically disallowed region. So if amino acid lies here in the white region, we will not accept that, because this is disallowed region for all amino acids except for glycine. So simply it can be drawn like this. If we have a sequence, if we have an amino acid sequence, you can see a sequence here. So the dots will be here. So these will be the angles between psi and phi, and they will be represented in dots. So
सर येलो पोर्शन तो डिसअलाउड रीजन है ना, तो वहां पे क्यों बन रहा है?
Wait for a while. So if we have like this protein, our protein is like this. So we need to determine, we need to know the white region should not have any bond, any dots. If there are dots in the white region, we need to know which white region, which dots are there. If that is for the glycine, that is okay. But if that is for other amino acids, we will reject that model, that protein structure. So, but if the angles, if the psi and bias, the phi and psi bonds and their dots occur in the yellow region, that is the permissible region, the outer limit, that is permissible. And what actually the red region, they are the allowed regions. So both red and yellow regions are okay for us. but these dots should not go out of in the white region. In the white region means that could only be glycine, otherwise the model is not correct. If that is glycine, that's okay, but if that is not glycine, we will reject the model.
Excuse me,
Am I
sir.
clear? जी.
सर ये येलो रीजन के अंदर एक बार दोबारा बताएं कौन से अमीनो एसिड आएंगे?
येलो रीजन्स के अंदर कोई भी अमीनो एसिड्स आ सकते हैं. दैट इज द आउटर लिमिट.
तो यहाँ पर कंफर्मेशन नहीं होगी अल्फा या बीटा की?
यहाँ पे कंफर्मेशन होगी ना, यहाँ पे कंफर्मेशन होगी. यहाँ पे देखें ये अल्फा हैंडेड अल्फा लेफ्ट हैंडेड अल्फा हीलिक्स है यहाँ पे, ठीक है. वो यहाँ पे आ जाएंगे, बीटा शीट्स यहाँ पे आ जाएंगे, लेकिन these बीटा शीट्स will be like this will be the outer limit for that. The yellow region will be the outer limit for that.
ओके सर.
ठीक है
सर.
जी.
सर ये क्वेश्चन है एक दो के डिफरेंट अमीनो एसिड्स के दरमियान जो साई एंड फाई बॉन्ड हो गए, उनकी बॉन्ड लेंथ यानी स्पेसिफिक होगी कि जब फर्स्ट करें कि कोई अगर सिस्टीन के साथ कोई और बात है तो उसके दरमियान बॉन्ड लेंथ स्पेसिफिक होगी. तो हम जो डिफरेंट अमीनो एसिड की रेसिड्यू सीक्वेंस पता है, हम उसको रामाचंद्रन प्लॉट में एंटर करते हैं, उस तरह प्लॉट सामने आ जाता है. तो हम उसकी बेसिस पे देखते हैं कि हमारे अमीनो एसिड रेड रीजन में है या येलो रीजन में, उसकी बेस पे हम बताते हैं कि बीटा शीट है या अल्फा हेलिक्स है.
एग्जैक्टली इसी तरह बिल्कुल इसी तरह.
ओके सर.
सर बेसिकली ये ग्राफ जो है वो कंफर्मेशन पे डिपेंड करता है एंगल की.
हाँ ये हाँ देखें अब हम we can discuss it now. So if the phi angle is 60, if that is 60, the phi angle अगर हमारे पास 60 है, 60 डिग्रीज है, ठीक है, और psi angle जो है वो भी 60 है, वो प्लस 60 यहाँ पे है, ठीक है. So phi angle and psi angle, so here confirmation हम ये कह सकते हैं कि वो confirmation कौन सी बनेगी? That will be left-handed alpha helix. That will be here. But if that is 0, अगर psi angle 0 है और phi angle 0 है, so that will be here. So that will be disallowed. समझ आ रही है?
Yes sir.
So from here, from this psi and phi, we can determine the protein conformation. Here, that is, for instance, 150, and this phi is actually, if you can see, that is minus 100. Like for instance, that is minus 100. So it will make beta sheets. Am I clear? आगे जाके सारी चीजें इसी की explanation है. Who will explain like up to now? जी फातिमा.
सर इसमें हम रामाचंद्रन प्लॉट में स्पेस फिलिंग मॉडल है पेप्टाइड का, और इसमें हम कैलकुलेट करते हैं फाई और साई रीजन की वैल्यूज को, और इससे हमें कंफर्मेशन पता चलती है प्रोटीन की. अब जो प्लॉट में हमारे पास रीजन्स हैं वो कुछ डिफाइंड रीजन्स है. जो रेड रीजन है वो बीटा शीट का होगा, और जो येलो अगर उसके ऊपर है तो वो उसकी आउटर लिमिट होगी, और जो व्हाइट रीजन है वो सिर्फ ग्लाइसिन के लिए और किसी और अमीनो एसिड के लिए नहीं है. तो इस तरह हमारी जो एंगल्स बनेंगी उससे हमें पता चलेगा कि वो बीटा शीट है या लेफ्ट हैंडेड है, राइट हैंडेड है. वो जिस एंगल पे आएगा तो इस तरह हमें पता चल जाएगा. और ये जो रीजन है ये डिफाइंड है, रीजन फिक्स रीजन है
हाँ ओके
प्लॉट के ऊपर.
ठीक है, थैंक्स. एंड नेक्स्ट इज हिजाब योर टर्न. एक्चुअली यू डिडंट शो अप.
इसमें भी जस्ट इस प्लॉट में ही बताया हुआ है स्पेस फिलिंग मॉडल के बारे में कि जोन से जगह मतलब स्पेस होगी उसको हम इस स्पेस फिलिंग मॉडल के थ्रू मतलब फिल कर लेंगे. और ये प्लॉट जो है हमें कंफर्मेशन के बारे में बताता है प्रोटीन्स की. और इस प्लॉट को हम लोग ड्रॉ करते हैं क्योंकि इसके कुछ डिफरेंट रीजन्स होते हैं. लाइक वाइट रीजन्स होते हैं, उसमें ग्लाइसिन के अलावा कोई और अमीनो एसिड मतलब नहीं अलाउड होता वहाँ पे. अगर वहाँ पे कोई और अमीनो एसिड होगा ग्लाइसिन के अलावा तो हम उसे डिसअलाउड रीजन कहेंगे. और रेड रीजन्स को हम अलाउड रीजन्स कहते हैं क्योंकि इसमें अल्फा हेलिकल्स और बीटा कंफर्मेशंस होती हैं. और अगर येलो रीजन में कोई मतलब अमाइनो एसिड आएगा तो उसे हम आउटर लिमिट कहेंगे।
ठीक हो गया। अच्छा कलीमुल्ला आपको समझ आ रही है? कलीमुल्ला
यस सर।
ठीक हो गया। एंड फायका फातिमा, फायका आपको समझ आ रही है?
यस सर आगे।
चलें, that's good. So moving further, if you can see now this is actually the result of what we were talking previously. Here, now we have previously talked about this. These are the beta sheets in this region. So now we can further like divide this beta sheet into conformation. If that is here, that will be anti-parallel beta sheets. Here, there will be parallel beta sheet if that lies here. Here, that will be collagen triple helix. So there will be collagen triple helix. This will be a bit different from the normal beta sheets. So, and that is here the right twisted beta sheets that these will lies here, that is has been encircled in the dotted lines with dotted lines. And here the alpha helix, the left-handed alpha helix, here that is right-handed alpha helix, here that will occur like here. So, and this will be alpha, this will be the phi, and this will be the psi degrees. So this is how it could be represented like this in this diagram as well. So due to high abundance of glycine and proline content in the collagen में glycine or proline, they are more in collagen, and due to the high content of proline and glycine, and we already have talked about this, that glycine and proline, they will not behave like normal amino acids. So in the collagen they are more, and that's why collagen fails to form a regular alpha helix or beta sheets, and they will not be producing a normal regular alpha helix and beta sheets, and therefore they will be forming three left-handed helical strands twisted to form right-handed triple helix. And a collagen triple helix has 3.3 residues per turn. In every turn, there will be 3.3 residues per turn. Am I clear up till now?
सर
जी
सर इस ग्राफ के अंदर ये फिक्स है कि इन एंगल की इन वैल्यूज के ऊपर यहाँ पर यही एंगल्स बनेंगे, यही स्ट्रक्चर्स बनेंगे।
एग्जैक्टली ये नॉन है, असल में ये देखा गया कि इसी तरह होंगे ये। जी जी। And from now, if our bases are like now a bit more mature, and that is the going towards Ramachandran principle, and is the alpha helices and beta sheets and turns, they are almost likely conformations for a polypeptide chain to adopt. So now we have, from based on the Ramachandran plot, there are only three conformations possible, and the first one is the alpha helix, the beta sheets and turns, So they are the possible conformations within a peptide chain, because most of other conformations are impossible due to steric collision between atoms. Because of the steric collisions between atoms, no other conformation is possible. And phi and psi dihedral angles, they determine the conformation of a polypeptide. So the polypeptide chain, within the polypeptide chain, these phi and psi, the dihedral angle between, so that will mean the angle between the two planes, that determine actually the conformation of a polypeptide chain. So we have previously talked about this, like there are three conformations that are possible. Actually two conformations, the alpha helices and the beta strands, and along with that that beta strands are there, and they are due to the two and the dihedral angles, and that is determined by polypeptide, the conformation of the polypeptide. You can see the two planes are there, and these two planes actually that determines the dihedral angle. Am I audible?
Yes, sir.
Okay, okay. So you can see we already have discussed about the amino acid, and the amino acid have a chiral carbon, and this one is the alpha carbon, that is the chiral carbon, and this is the alkyl group, this is the amino group, and this one is the carbon, the carboxyl carbonyl carbon. So if you can see, this one will be in the one plane, this one will be in the second plane, and the angle between them will be dihedral angle. But how to determine phi and psi angles, the angle between the bond, the bond that makes an angle, and that the phi bond and the psi bond between carbon-carbon and between carbon-nitrogen, and how we can determine psi angle and phi angle, how we can determine. Let's talk about them. So before that, here at the Ramachandran plot, you can see each dot shows a pair of psi and phi dihedral angles that occur in a protein. So each dot will represent a pair of the angles like this phi and psi. And where there is no dot, that will mean there is no the dihedral angle, means that there will be no amino acid. So much of the amino acids, much of these angles, they lie here at this position, that will be the beta, at this position that's alpha, and at this position. And rest of the white region, if we model a protein and the dots are placed here, so that protein structure will not be like reliable. And if our protein dots, the dots of a protein that lies here, that means that is acceptable model and that can rely upon. So dot clustered into alpha helical and beta strands and turns at right conformations, and the conformation with no dot do not occur. And here, this diagram, there are 100,000 dots. These are 100,000 dots taken from high resolution crystallographic structure. So that means from where we can determine the structure from crystallography, that is common practice. NMR is the second. So crystallography, X-ray crystallography is the main source of getting the structure. So further, the dihedral angles are defined by the positions of 4 atoms covalently bonded in a series. So we need to determine the position of 4 atoms, and those 4 atoms will give us the angle like phi and psi. So to determine the phi and psi, we need to position 4 atoms. So this is important. Like we need to position these 4 atoms, and that will give us the phi and psi angle, and that's it. So for determining phi and psi angle, again, we need to know the position of 4 atoms. Am I clear?
Yes, sir.
Okay.
सर एक दफा रिपीट कर दें.
सो अगर हम कमिंग स्लाइड पे चले जाएं तो ये सारी डिटेल उसमें है. सो जस्ट मेरा मतलब यहाँ पे ये है कि फाई एंड साई एंगल को डिटरमिन करने के लिए हमें ये किस तरह पता चलेगा कि ये फाई एंगल लाइक दैट्स -180, -90, 100 और 180 एंगल क्या है इसका फाई, और साई एंगल क्या है. सो टू डिटरमिन दोज वी नीड टू हैव 4 atoms positioned in a polypeptide. So हमें वो 4 atoms मालूम होने चाहिए, उसके बाद हम इन angles को मालूम कर सकते हैं. So let's see how we can determine. And one more thing, sorry, preproline, that's a residue preceding a proline. Before this, we have already talked about this, that in this white area, only glycine can be the possible conformation. So glycine can lie there in the white region, or proline can be there, and these two amino acids like glycine and proline, they do not behave like normal amino acids, and you know better than me why they don't behave like normal amino acid. The first, for glycine, because glycine does not have a chiral carbon, and proline, for proline, its side residue, the alkyl group, is being like bonded with the amino group. So that's not a normal amino acid. And pre-proline, like the proline, wait, like we have a proline amino acid in a peptide chain. So this proline will behave differently from the other amino acids. But pre-proline, like there is methionine before proline. So the proline, the alkyl group, is being attached with the amino group. So this pre-proline will also be having differently from normal proline, from normal amino acid. So this pre-proline is also important. So we will be checking three residues. The first one is glycine, the second one is proline, the third one is pre-proline, means the residue preceding proline. So these three residues are important in determining a protein structure. So if you can see this diagram and this animation moving, that means you can observe that, and you can see here the alpha carbon. The alpha carbon here, that is here, this is the carbonyl carbon. The white are hydrogen. The blue one is nitrogen. The red one is oxygen. And the bond, the covalent, the normal covalent bonds are shown in white color. And here the peptide bonds, the partial double-stranded bonds, that are shown in purple. So they are peptide bonds shown in purple. So the dotted structure is a structure of alanine. So alanine only has CH3 as a side group. Here is the alpha carbon, and with the carbonyl carbon here, and that is been attached with another amino acids, making a peptide bond. Here it is making a peptide bond. So this amino acid is being shown like this one as well, CH3, NH2, and this carboxyl carbon. Am I clear up till now?
Yes, sir.
So now how to determine the angles now, like the angle between carbon and nitrogen, and between carbon and carbon, that is the Psi bond, and this one is Phi bond. So how to determine the angles now? And why we need to determine? Because we need to determine the structure, like either it will be making beta sheets or alpha helices. So how we can determine? We can only determine by determining the angles. And here we need to determine the angles as well. So that's the question. So here in this diagram, again, the same thing. This is the Psi bond, and this one is the Phi bond. So each amino acid has two main chain backbone bonds that can rotate, defining dihedral angle. So this can rotate around it. So if you can see, this one can rotate, and this can rotate. So this angle can rotate, and this can rotate. So these two angles can rotate, and this will rotate the whole, like if it is here, so this can be like this one as well. So this can move if this bond rotate, this whole structure, if that is a polypeptide structure, this whole polypeptide structure can move from there to there and changing the whole protein conformation. Similarly for this one, this can change from here. This peptide bond can change from here, and the whole the polypeptide chain, if that is a polypeptide chain, that can move from here to here. So the whole protein structure is dependent on these two angles. So now, if you can see, the peptide bonds can, and these peptide bonds, they cannot rotate because they are partial double bonds. So the partial double bonds, they will not move. There is less tendency for them to move. Only the Phi and Psi bonds, they are flexible enough to move the whole conformation of the protein. So again, now let's check how we can determine Phi angle. So here the animation can be easy to know. Like previously we have talked about, we have to locate the 4 residues. This one is now the residue 1. This one is the nitrogen, and that is the residue 2. This one is the carbonyl carbon. This one is the alpha carbon, and this one is the residue 3, and this one is the residue 4. So keep in mind the angle, the bond which we need to determine will be in the middle of that. So if we want to determine the Phi bond, so let's keep it in the middle. So this one is the first residue, second residue, third residue, and fourth residue. Once we locate the residues, so now we can determine the angle. So how we can determine the angle? Check these 2 bonds. If they are making 0 angle between them, like they can overlap over one another. They are overlapping. So this will be 0 angle. So now the Phi is 0. Am I clear?
सर अगर ओवरलैप एंगल 0 बना रहा है तो 5 बॉन्ड बनेगा, यही बताया आपने?
Yes, exactly. So Phi bond 0 hoga. Phi will be equal to 0 because these 2 bonds are making 0 angle with one another. So the Phi bond will be 0, and the Phi is between nitrogen and the alpha carbon. So next is, if Phi is equal to 0, how we can determine? Move it is in the clockwise rotation. And now if it is in this conformation, this is 90 degree, Phi is 90 degree. If this is like this, so these 2 bonds are making 90 degrees. If that is like here, that is minus 180. See, that's minus 180 because they are like that. Like check it once more. That's 0 clockwise rotation. Here now 90. (180). So this is 180 degrees. एक दफा फिर देख लें. If you have any question in mind, let me know. So clockwise इसको अगर rotate करेंगे तो 0 to 180 degrees होगा. अगर anti clockwise rotate करेंगे या counter clockwise rotate करेंगे, so that will be the minus degrees. Minus 0, minus 10, minus 20, and so on. समझ आ रही कि नहीं?
जी सर आ गई.
आ गई है? Easy है? Easy है?
Yes sir.
That's good. And next to it is that was 0. So counterclockwise rotation starting at 0 gives negative dihedral angles from -1 through 90, ending at -180, and the same dihedral angle as +180. And check it like this. This is anti-clockwise or counterclockwise. That will give us negative values of the angle. It's minus, so check it once more. So this is minus 90, and on this side that was plus 90. So the bottom line is how we can determine an angle. First, we have to locate the 4 residues. Am I clear with that now?
Yes, sir.
Huh. So now how to determine psi angle? That was phi. So to determine the, okay, so let's check it in a plane. So in a protein sequence, they are be like this. This is in the one plane, this one is will be in the another plane, and this psi angle can move. So this will change the whole plane, and this phi can move, this will change the whole plane. So because they cannot rotate, each peptide bond hold 6 atoms in a plane. So this will hold 6 atoms in a plane. 1, 2, 3, 4, 5, and 6. This will be in the one plane, and these 6 will be in the another plane. So by moving this bond will change the position of the 6 atoms actually in the plane. However, most possible angles of phi and psi are impossible due to clashes between atoms. So most of the angles are not possible because these atoms will clash between one another due to their sizes. These balls are represented as much smaller than the atoms they actually represents. So these atoms are actually much larger than what has been shown. So if you can see, this is the actual size of these atoms. So if you can see this diagram, you will observe that actually less of the movement is being possible than previously what we have described. So this is less flexibility is being possible in this diagram. So this is not like as much flexible as being determined because the size of these atoms are much more. So if you can see, if this phi angle moves, the clashes are here. These are the clashes. Now this is okay. This is okay. Now this clash occurs. So that means that that cannot move from here in this position. So here it can move now, and now it's cannot move from here to from there, and so the clashes will occur if it moves more. So when psi is fixed at 165, so we need to fix one of the bond, the other will move, and we can know the clashes. So when psi is fixed at 165, if we fix psi at 165, most phi angles produces clashes. So if psi is fixed, phi will make clashes at most angles. This simulation displays clash. Here is a clash while permitting van der Waals overlaps that would be physically impossible. So let's see if psi is 165. Let's see in the Ramachandran plot. Psi is 165 here. So psi is 165 here. So here is psi is 160. Sorry, here is psi is 165. So what about phi? So if psi is 165, most phi angles produces clashes. So phi angles will produces clashes, most of the phi angles, except for this region, So they will produce clashes at this position. So here, at this position, they will produce clashes. So there will be clashes here, and they cannot, because and that's why this region is white. And next to it is this simulation displays clashes while permitting one. Okay, we are done with that. Okay, now here you can see at minus 165 psi is minus 165. So, and that is fixed at 165. Clashes occur at phi angles except minus 70. Minus 70 is here, except as minus 70 phi to minus 170. They are possible for phi, and the rest are not possible. And this range, this possibility range, is beta strained region. And like you can see the conformation as well, the movement of the amino, the peptide bond. Sorry, the phi bond. So if phi bond moves, it will produce clashes at with the movement, and only these conformations are possible because there will be no clashes here. And here in the above animation, phi is fixed at minus 80. So phi is fixed. Now the turn is for the psi. We need to determine the psi. If phi is fixed at minus 80, now check how much conformation of psi is possible, how much angles of psi is possible. So here you can see the two conformations of psi is possible. One range from plus 10 to minus 55. So here plus 10 to minus 55, that is possible, and that is the alpha helix conformation. And the second one is plus 90 to 180. This one is possible. So these two conformations are possible, the beta and the alpha, and that is at minus 85. So by fixing phi at minus 80, psi can range in two possibilities, this and this one. These two ranges are given. And next to it is, if phi is made fixed at 0, if phi is fixed at 0, clashes at all psi angles will occur. So clashes here, clashes at all psi angles will be there. So you can see clash start from here, when it ends, the clash start from there. So at all angles of phi, at all angles of psi, there will be clashes if phi is kept 0. And that's it. इसकी समझ आ गई है?
Yes, sir.
Am I clear? Who will explain the whole thing? Who will explain the whole thing?
सर मैं कर दूं?
जी प्लीज.
सर हमने असल में लास्ट टाइम देखा था कि फाइव बॉन्ड होते हैं प्रेजेंट, जिसके ऊपर क्या नाम है जो डिफरेंट प्लेन पे क्या नाम है एंगल रोटेशन होती है. तो हम इसमें रामाचंद्रन प्लॉट के थ्रू और देखने लगे कि फाई की और साई की कितने एंगल पे जो हो रही है रोटेशन हो रही है. तो सबसे पहले तो हमने ये डिस्कस किया था फाई एंड साई दोनों डाइहेड्रल बॉन्ड्स हैं. तो अगर हमें इनकी एंगल मालूम करना है तो हमें 4 जो रेजिड्यूज हैं या 4 एटम्स हैं उनको पहले पोजीशन आउट करना पड़ेगा। उनकी पोजीशन के बेसिस के ऊपर फिर हम उसके बाद बता सकते हैं कि फाई कौन सा बॉन्ड बना रहा है और साई कौन सा कितना बॉन्ड एंगल बना रहा है और साई कितना बॉन्ड एंगल बना रहा है। तो सबसे पहले हमने जो देखा कि हमने पहले जो 4 रेजिड्यूज थे उनको लोकेट आउट किया 1 2 3 4। अगर फर्स्ट एंड 4 आपस में ओवरलैप कर रहे हो तो उस टाइम जो फाई का जो सॉरी साई का जो बॉन्ड होगा बॉन्ड एंगल होगा वो 0 होगा। और इसी तरह अगर हम उसको क्लॉकवाइज मूव करवाते जाएंगे तो 90 डिग्री बनेगा, पॉजिटिव प्लस 90 डिग्री बनेगा एंड इस तरह प्लस 80 डिग्री बनेगा। अगर उसको हम एंटी क्लॉकवाइज मूव करवाएंगे तो फिर वो नेगेटिव में चला जाएगा, यानी नेगेटिव 90 डिग्री एंड नेगेटिव 180 डिग्री। और इसके बाद हमने ये चीज डिस्कस की थी कि जो हमने पहले पिक्चर्स देखी थी या ग्राफ फोटोग्राफ्स देखे थे उसमें जो एटम्स थे वो छोटे शो किए थे, लेकिन एक्चुअली में जो एटम्स होते हैं वो स्मॉल नहीं होते। तो ड्यूरिंग रोटेशन इन एटम्स के जो दूसरे लिंक एटम्स होते हैं, जो हमने बात की कि एक प्लेन के अंदर 6 एटम्स, जो एक जो 5 बॉन्ड है या साई बॉन्ड है वो 6 एटम्स को अपने साथ क्या नाम है रिवॉल्व करवाता है। तो ये अगर एटम्स का साइज बड़ा होता है तो इनकी वजह से इनकी आपस में क्लैशेस होती हैं। तो वो क्लैशेस को हम किस तरह विजुअलाइज कर लेते हैं, रामाचंद्रन प्लॉट के थ्रू हम विजुअलाइज कर सकते हैं। तो इसी जो स्लाइड के ऊपर अगर हम देखते हैं कि अगर हम 5 बॉन्ड को फिक्स करवा देते हैं 0 डिग्री सेंटीग्रेड पे 0 डिग्री एंगल पे, तो उसके बाद जितने भी साई बॉन्ड बनेंगे हम रामाचंद्रन प्लॉट के अंदर देखते हैं कि उसको कोई भी जो वाइट जोन के अंदर कोई भी अमाइनो रेजिड्यू नहीं प्रेजेंट है, तो उसका मतलब है कि जितने भी एंगल जो साई बॉन्ड बनाएगा उसके ऊपर क्लैशेस आएंगे। इसी तरह हमने 165 डिग्री पे देखा था कि अगर हम साई को 160 डिग्री पे फिक्स करवा देंगे तो फाई जो है वो सिर्फ -70 से 170 के दरमियान जो है क्लैशेस नहीं देगा, जो फॉल करता था अल्फा और बीटा डोमेन्स के दरमियान यानी रीजन्स के दरमियान। बाकी इसके अलावा जितने भी एंगल्स होंगे वो आपस में क्लैश करेंगे।
Well done, great. Great. So now one thing we need to know, like how to locate phi, psi and phi angles. So to determine the psi, and this will be the first atom, the first residue atom, this will be the second one, this will be the third, and this will be the fourth. And to locate the phi, this will be the phi between carbon and nitrogen, and this one is the carbon. So this is the psi and this is the phi. So to determine the phi, this will be, when this atom will be the first, second, third, and fourth. That will mean we will be making that very specific bond in the middle. Like if we locate psi, this will be the one, the second, the third, and the fourth. And similarly is for this one. So for this will be the 1, 2, and third and fourth. Am I clear?
Yes, sir.
Okay, who will explain again, like the whole, who will summarize the today's talk? By the way, Shafiq did it very well, but I want some other person as well to know either everybody is clear or not. Ji, Rabia.
सर इसमें आपने सबसे पहले ये बताया कि रामचंद्रन प्लॉट के अंदर जितने भी डॉट्स होते हैं वो रिप्रेजेंट करते हैं कि यहाँ पाई और साई एंगल्स हैं, यानी अमाइनो एसिड्स हैं, और जो ब्लैंक एरिया होते हैं वहाँ रिप्रेजेंट करते हैं कि यहाँ एंगल्स नहीं है। उसके बाद जो है ये फाई एंड साई एंगल डाइहेड्रल एंगल्स होते हैं, यानी इन एंगल्स को रिप्रेजेंट करने के लिए 4 एटम्स रिक्वायर्ड होते हैं। फाई एंगल्स को जब रिप्रेजेंट करते हैं तो इसके अंदर ये जो है ये कार्बोनाइल अल्फा कार्बन और नाइट्रोजन के दरमियान में बॉन्ड बनता है। और इसमें किस तरह से होता है कि जब ये 0 डिग्री पे एंगल होता है और जब इसे हम क्लॉकवाइज मूव करवाते हैं तो ये 90 की तरफ जाता है और 90 से फिर 180 की तरफ जाता है। और अगर इसे एंटी क्लॉकवाइज मूव करवाएंगे तो ये -90 की तरफ से -180 तक जाएगा। और इसी तरह साई एंगल्स में भी इसी तरह है। लेकिन अगर हम जो है वो फाई एंगल को फिक्स कर देते हैं, फिक्स करने से जो है वो फिर साई एंगल के अंदर जो है वो क्लैशेस आना शुरू हो जाते हैं। और इसी तरह अगर हम सबसे पहले साई एंगल को अगर हम फिक्स करते हैं 165 के ऊपर, तो फाई एंगल जो है वो सिर्फ -70 से -170 तक उसकी जो लोकेशन है वो एक्सेप्टेबल है। इसके अलावा वो कहीं पे भी जो है वो एक्सेप्टेबल नहीं होगी, और वो जो है वो बीटा स्ट्रैंड्स के अंदर आता है। और अगर हम फाई एंगल को फिक्स करते हैं 0 के ऊपर, तो इसमें जो है वो साई एंगल क्लैशेस देते हैं यहाँ पर एक और भी चीज थी कि यहाँ पर अगर हम साई एंगल को एक और जगह फिक्स करते हैं तो वहां पर जो है वो साई फाई एंगल जो है वो दो जगह पे उसकी पॉसिबिलिटी है। जैसे यहाँ पर अल्फा स्ट्रैंड्स के अंदर आपने बताया था कि 10 से जो है वो -55 की तरफ है, और इसके अलावा एक और जो थी लोकेशन थी वो 10 से अबव थी, वहां पर एक्सेप्टेबल है जो अल्फा हेलिक्स के अंदर लोकेशन आ रही होती है।
जी, great, great. One more person who could explain. Who will explain? By the way, these two, Rabia Jawad and Shafiq, they did it well. Majidur Rahman. मजीद आवाज आ रही है आपको? मायदा, Maida is there, Maida? Faika, Faika. Faika, can you hear me? जी, Faika. Okay. फाइजा आप बता दें, फाइका की आवाज नहीं आ रही। फाइजा?
जी सर।
जी, could you explain?
जी
इसको
सर,
एक्सप्लेन,
आपने
जी जी,
आज
इसको समझ लें बिकॉज आगे जाके जब हम प्रोटीन कन्फर्मेशन पे जाएंगे, सो दीज आर द बेसिस फॉर दैट। ठीक है, सो प्लीज आप एक्सप्लेन करें इसको क्या। जी जी
सर, आपने डाईहेड्रल एंगल्स के बारे में बताया कि साई और फाई दो एंगल्स हैं, और इनको अगर हमने पोजीशन करना है तो हमें सबसे पहले 4 एटम्स जो होंगे उनको पोजीशन करना होगा सीरीज में। वाइट एरिया आपने बताया था कि वो एरिया होता है जहाँ पर ग्लाइसिन और जो प्रोलीन है सिर्फ वही आ सकते हैं, क्योंकि ये जो है वो डिफरेंटली बिहेव करते हैं दूसरे अमाइनो एसिड से इनके स्ट्रक्चर की वजह से। और प्री प्रोलीन का आपने बताया था कि ये प्री प्रोलीन भी इनके ग्लाइसिन और प्रोलीन के अलावा एक और रेजिड्यू है जो कि प्रोलीन से प्रीसीडिंग रेजिड्यू होता है। और जो उसके बाद आपने जो है वो साई और फाई को का बताया कि इसको किस तरह से ये रोटेट कर सकते हैं, जो जबकि जो पेप्टाइड बॉन्ड्स होते हैं वो रोटेट नहीं कर सकते। और फिर अगर हमने डिटरमाइन करना है कि किस तरह से पाई बॉन्ड रोटेट करता है, तो पाई बॉन्ड की रोटेशन होती है, अगर वो पहले हमने देखा था कि उसका एंगल जो है वो 0 है, और अगर 0 से उसको हम रोटेट करना शुरू करें क्लॉकवाइज तो वो जो है वो एंगल आता है 90 और फिर उसके बाद 180 पॉजिटिव एंगल आता है। लेकिन अगर उसको हम एंटी क्लॉकवाइज रोटेट करें तो वो फिर नेगेटिव वैल्यूज हमें देगा 0 से -180 तक। उसके बाद हमने साई की डिटर्मिनेशन देखी कि हम साई के एंगल को किस तरह से डिटरमाइन करते हैं। इसके लिए आपने बताया था कि प्लेन में 6 एटम्स होने चाहिए, और जो कि जब हम उन्हें मूव करते हैं तो वो अपनी पोजीशन को चेंज करते हैं। और फिर आपने ये बताया था कि अगर हम क्लैशेस को देखें, कि अगर हम फिक्स रखें फाई बॉन्ड को, तो जो साई बॉन्ड होता है वहां पर 70 से 170 तक हमारे पास उसमें आते हैं, उसका एंगल आता है। और जो पाई बॉन्ड होता है, अगर हमने उसकी कंफर्मेशन, उसमें 2 कंफर्मेशन आती है, एक अल्फा और एक बीटा। जो अल्फा होती है वो +10 से -55 तक आती है, और जो बीटा है वो +90 से +80 तक होती है।
जी जी, ठीक है,
सर ऐसे ही है।
जी जी, बिल्कुल ऐसे ही है। बस जस्ट अब बॉन्ड एंगल को डिटरमिन करना रह गया कि हाउ वी कैन डिटरमिन द एंगल, द बॉन्ड एक्चुअली, कि उसको हम एटम्स को किस तरह डिटरमिन करेंगे। लेकिन हमें किस तरह पता चलेगा कि दिस इज फाई एंगल एंड दिस इज साई एंगल।
सर, अगर हमने 165 के ऊपर लोकेट किया है फाई को, तो जो साई को, तो जो फाई होगा वो क्लैशेस बनाएगा।
जी जी, नहीं मैं ये कह रहा हूँ कि हाउ वी कुड नो द पोजीशन ऑफ दिस साई बाय डिटरमिनिंग द पोजीशन ऑफ दीस 4 एटम्स। ठीक है? Like if we want to determine determine to
से
determine
फोर
psi,
अगर एटम
अगर हम
अगर
हाँ अगर हम psi को determine करना चाह रहे हैं, so this one is the psi bond. So उस केस में हम this will be the number one atom, this will be the second, this will be the third, and this one will be the fourth. This one will be the fourth. और अगर हम इस phi bond को करना चाह रहे हैं, so this one will be the first, and this will be the second, and this one will be the third, and this one will be the fourth. Am I clear?
यस सर
Yes, sir.
चलो. And okay, and next to it is classes of proteins. We have different classes of proteins, and we can determine those on the basis of function and structure. Based on the function, we have different types of those, like enzymes. They accelerate biochemical reactions. Structural, some proteins are structural proteins, and they form biological structures. Some work in transport. They carry biochemically important substances. Some are in the defense mechanisms. They protect the body from the foreign invaders. So these are the classes of proteins that we can classify based on the function. And next is some of the proteins can be classified on the structure, like they form globular structure, and that is a complex folds, irregular shaped tertiary structures. Fibrous, some proteins are fibrous. They could be extended, they could be simple folds, generally structured proteins. So some proteins so the proteins can be classified on the cellular localization definition. So we can localize them as a part of the membrane, indirect physical contact with a membrane, generally water insoluble. Some are soluble in water, like they will be soluble in water, and they can be available anywhere in the cell. So these are the three classes, three main categorization of the proteins. So this categorization is on the base of function. This categorization is the based on structure, and this one is the cellular localization. And we will talk about the rest in different techniques, like we can predict structure through bioinformatics, as well as we can do that with experimental approaches. And actually, through experimental approaches, we got some data, and based on that data, we can predict the protein structure computationally, like through bioinformatics. So in experimental techniques, first of the technique that is too much famous and too much reliable is X-ray crystallography. And X-ray crystallography, this means this will be using X-rays and crystal structure. That will mean it will require crystal structure to determine the structure of the protein. Am I clear?
यस सर
Okay, so, and this technique actually determines three-dimensional molecular structure of a crystal. The method requires crystal, and they need to be of certain quality and size. So, so the quality and size of the crystals are required. But there is a problem, and the problem is to make crystal structure from salt is easy, but producing crystals from complex molecules like protein is often met with failure. There are certain proteins that could not be crystallized. So there are limitations with this procedure, this X-ray crystallography. There is another technique, and that is NMR, nuclear magnetic resonance. So this technique determines what this determines the quantity, the quality, and the protein structure of a sample. So the NMR is a technique, but this required small peptides. This could not be used for larger proteins. So there are limitations in this procedure. There are limitations in this procedure as well. And due to these reasons, there are limited number of protein structures that are resolved so far. So the limitations of the current experimental procedures or techniques is like this can be determined by the data from the data. Like in the protein data bank, this is a data bank that contains the crystallized structure. The resolved crystallized structure are deposited in PDB. So there are more than 23,000 protein structures. This is an old data, by the way. There are more than 23,000 protein structures present in the PDB. While in Swiss prot, there are 100,000 of proteins. So you can imagine how much less number of protein structures are resolved. And we do have the structures available. And non-redundant proteins means the proteins that are not repeated, they are more than 10,100,000. So in 10,100,000, only 23,000 protein structures are available. So this shows the limitations of the current experimental techniques like the NMR and X-ray crystallography. So the importance of structure prediction, this will fill the gap is to fill the gap between sequence and structure. So there lies a gap between the two, the sequence and the structure. So there is a need to fill the gap. For protein engineering, if we want to alter a protein structure, what will be what will be the effect on the function? So for that reason, we also need to predict the structure. So if we want to alter the function, we need to predict the structure. And third is in the drug design, we also need protein structure. So these are the areas where we need protein structure. So computer simulated based on energy calculation. So we can predict techniques. There are techniques through which we can simulate the structure of a protein. And the first of the method and the group of methods is on energy calculation. So based on energy calculation, we can predict the protein structure. And those may be physiochemical principles, thermodynamic equilibrium with minimum free energy. This is another of the technique. Global minimum free energy of protein surface. So these are computationally simulated procedures to predict structure through energy calculation. Alternatively, we have some other techniques, and that group is known as knowledge-based approaches. So we have some prior information based on that information, we can predict the structure. And the first of the method is homology based method. We already know some protein structure, and by having homologous sequence means the sequence like that, the one for which the sequence is the structure is available. So that means a homologous sequence or similar sequence will give rise to similar structure. So if we have two sequences and one sequence the structure is available, and for the other sequence, for like I have another sequence, and for that the structure is not available. So if that is not available and these two sequences are homologous, means if they are 90% identical or more than 90% identical, so that means that will tend to give rise to the same structure. So that can give rise to the same structure. So through the homology, we can predict the structure. And the next of the knowledge-based approaches is threading approach. Threading approach is actually the fold recognition method. Like if a sequence, if I have a sequence and I know like this sequence has certain folds, and like I have another sequence, and in that sequence I have the same homology of in this region, so it will give rise to this fold, and for this fold some another region for that fold, and some another region for the third fold. So this is actually threading approach. Sorry class. Okay, so threading is another technique, and through this technique, we can predict the protein structure. And third of the method is hierarchical method, and this approach actually combines some of the procedures in the homology method and some of the techniques that are using in threaded matting method. It combines the two methods, and these are actually hierarchical methods that they are a bit intelligent way of protein modeling. And in the homology modeling, okay, class यहाँ तक समझ आ गई है?
Yes, sir.
Okay, who will like explain up till now what we have covered?
Sir, ma'am.
जी शफीक.
सर हमने डिफरेंट अप्रोचेस देखी बायोइन्फॉर्मेटिक्स से डिसाइड हम कुछ अप्रोचेस देख रहे हैं उनके थ्रू हम क्या नाम है प्रोटीन स्ट्रक्चर प्रिडिक्ट कर सकते हैं. तो सबसे पहले हमने एक्सरे क्रिस्टलोग्राफी देखी कि उसके जरिए हम एक्सरे जो एक्सरे बम्बार्ड करके 3D स्ट्रक्चर ले लेते हैं प्रोटीन का, लेकिन इट इज सो स्पेसिफिक फॉर द क्रिस्टल लेटिस. अगर उसके अलावा अगर कॉम्प्लेक्स स्ट्रक्चर आ जाता है, ज्यादा क्रिस्टल कॉम्प्लेक्स हो जाता है, तो उस उधर क्या नाम है उसकी लिमिटेशंस आ जाती है. इसके बाद MMRI था, मैग्नेटिक जो था, उसके अंदर भी इसी तरह था कि हमें एक स्पेसिफिक स्ट्रक्चर मिल जाता है, लेकिन उसकी भी सम लिमिटेशंस थी मॉलिक्यूलर स्ट्रक्चर. इसके बाद हमने कुछ जो टेम्परेचर बेस्ड ये एक फिर हमने देखा कि टोटल कितने हमारे पास प्रोटीन स्ट्रक्चर अवेलेबल है. तो 100,000 जो प्रोटीन नॉन रिडंडेंट प्रोटीन स्ट्रक्चर्स वो अवेलेबल है, लेकिन उनमें से ओनली 23,000 एक्चुअल 3D प्रोटीन स्ट्रक्चर प्रोटीन डाटाबेस पे अवेलेबल है. इस और इम्पोर्टेंस ऑफ स्ट्रक्चर एंड प्रेडिक्शन में हम सीक्वेंस और स्ट्रक्चर, जो प्रोटीन का स्ट्रक्चर होता है वो सीक्वेंस और स्ट्रक्चर के दरमियान का जो गैप होता है वो फिल कर देता है. ये हमें इस पर हेल्प आउट करता है जब हमें कभी किसी प्रोटीन के फंक्शन को ऑल्टर करना हो, तो हमें पता हो कि हम कौन कौन से सीक्वेंस को चेंज करेंगे तो उस प्रोटीन का ये वाला फंक्शन ऑल्टर हो जाएगा. ये हमें हेल्प आउट करता है ड्रग डिजाइनिंग में. इसके बाद हमने जो दूसरी टेक्निक थी टेम्परेचर बेस्ड टेक्निक, उनमें हमने देखा था जो फिजियो केमिकल प्रिंसिपल्स थे, थर्मोडायनामिक इक्विलिब्रियम और प्रोटीन सरफेस के ऊपर जो डिफरेंट अप्रोचेस थी, यानी जो एनर्जी कैलकुलेशन, जो ओवर एनर्जी यील्ड था प्रोटीन के, उसके बेस के ऊपर भी हम प्रोटीन का स्ट्रक्चर क्या नाम डिटरमाइन कर सकते हैं. और फिर इसके बाद नॉलेज बेस्ड अप्रोचेस थी जिसमें होमोलॉजी फर्स्ट अप्रोच थी, जिसके अंदर हम होमोलॉजी में कहते हैं कि अगर हमारे पास एक सीक्वेंस ऑलरेडी मौजूद है जिसका हमें एक सीक्वेंस है उसका हमें प्रोटीन स्ट्रक्चर भी मालूम है, तो अगर हम किसी दूसरी सिमिलर प्रोटीन सीक्वेंस का अगर स्ट्रक्चर डिटरमाइन करना चाहिए तो हम उसके साथ होमोलॉजी के थ्रू उसको निकाल सकते हैं. इसी तरह थ्रेडिंग अप्रोच थी और जो हिराइकल अप्रोच थी वो इन दोनों का मिक्सचर थी.
ओके, दैट्स ग्रेट. ठीक है. ओके, हु विल एक्सप्लेन नेक्स्ट? कौन एक्सप्लेन करेगा? मायरा इज देयर. मायरा, फाइका ठीक है, फाइका बता दें.
सर इसके अंदर सबसे पहले हमने बात की थी कि प्रोटीन प्रोडक्शन की जो
प्रिडिक्शन है प्रोटीन स्ट्रक्चर की, उसके लिए हम कौन कौन सी टेक्निक्स यूज करते हैं। इसमें सबसे पहले हमने डिस्कस किया था एक्सरे क्रिस्टलोग्राफी का। इसके अंदर हमने देखा था कि हमें एक्सरे चाहिए और हमें क्रिस्टल्स चाहिए। क्रिस्टल्स जो होते हैं वो सॉल्ट क्रिस्टल्स को ग्रो करना तो बहुत ईजी होता है, लेकिन उसके जब हम क्रिस्टल्स का कॉम्प्लेक्स मॉलिक्यूल बना के प्रोटीन की तरह का रखते हैं तो उसके अक्सर औकात हमारे वो प्रोडिक्शंस गलत हो जाती हैं और ये इसकी लिमिटेशन आ जाती है। लेकिन इसका प्लस पॉइंट ये होता है कि हम इसमें 3D स्ट्रक्चर प्रिडिक्ट कर सकते होते हैं और जो क्वालिटी और साइज है वो हम प्रिडिक्ट कर सकते हैं। सेकंड जो हमने टेक्निक स्टडी की वो थी न्यूक्लियर मैग्नेटिक रेजोनेंस, NMR। NMR के अंदर हमें कंटेंट और प्योरिटी मिलती है और उसके अंदर जो स्ट्रक्चर हम प्रिडिक्ट करते हैं वो मॉलिक्यूलर स्ट्रक्चर होता है। मगर जो NMR होता है इसके अंदर हम बहुत ज्यादा बड़े बड़े प्रोटीन्स प्रिडिक्ट कर रहे होते हैं, तो इसके अंदर कॉम्प्लेक्सिटी बहुत ज्यादा हो जाती है, जिसकी वजह से कि हमारी एक्यूरेट रिजल्ट्स वगैरह डेविएट कर सकते हैं। हमारी कॉम्प्लेक्सिटी इसकी जो होती है वो लिमिटेशन में चली जाती है। अब अगर हम इससे आगे जब हमने लिमिटेशंस की बात की थी जो कि करंट एक्सपेरिमेंटल टेक्निक्स यूज हो रही हैं, उसके अंदर हमारे पास सबसे पहले था प्रोटीन डाटा बैंक। प्रोटीन डाटा बैंक जो था वो तकरीबन 23,000 प्रोटीन स्ट्रक्चर्स के लिए था, यानी कि इसमें हमारे पास स्ट्रक्चर्स बहुत कम है, इसकी क्वांटिटीज की लिमिटेशन में चली गई है। जो स्विस पोर्ट है वो तकरीबन अप्रोक्सीमेटली 1 लाख प्रोटीन्स के लिए होता है और जो नॉन रिडक्टेंट होता है वो के 10,000 प्रोटीन्स के लिए होता है, क्योंकि NR के अंदर प्रोटीन्स बहुत ज्यादा हैं, इसकी कॉम्प्लेक्सिटी इसकी लिमिटेशन चली जाएगी और प्रोटीन डाटा बैंक के अंदर बहुत थोड़े प्रोटीन्स हैं, इसलिए इसकी वो लिमिटेशन में चला जाएगा। अब सेकेंडली अगर हम इसकी बात करें कि हमें स्ट्रक्चर प्रिडिक्शन की जो होती है इसकी इंपॉर्टेंस क्या है कि हम प्रोटीन्स का स्ट्रक्चर क्यों प्रिडिक्ट कर रहे होंगे। हमारे पास जो प्रोटीन्स होते हैं उनके सीक्वेंस और उनके स्ट्रक्चर के बीच में जो डिफरेंस आ रहे होते हैं, जो गैप्स आ रहे होते हैं, उनके लिंक करने के लिए हमें प्रोटीन के स्ट्रक्चर चाहिए होते हैं। सेकेंडली जब हम प्रोटीन का स्ट्रक्चर प्रिडिक्ट कर लेंगे और हमें ये बात पता होगी कि कौन सा स्ट्रक्चर कौन से फंक्शन के लिए यूज हो रहा है, तो हम किसी भी प्रोटीन का फंक्शन ऑल्टर कर सकते हैं। जब हमें उसके पूरी होमोलॉजी वगैरह उसका पूरा स्ट्रक्चर पता लग जाएगा, हम अपनी मर्जी की उसके अंदर ऑल्टरेशंस वगैरह करनी हमारे लिए पॉसिबल हो जाएंगी। थर्ड पे हमने बात की थी ड्रग डिजाइन की। ड्रग डिजाइन करने के लिए हमें प्रोटीन्स चाहिए होते हैं और प्रोटीन्स के अंदर जब हम वेरिफिकेशन वगैरह कर सकते वेरिएशंस वगैरह कर सकते हैं तो हम अपनी मर्जी का ड्रग जो है वो इसके थ्रू ले सकते हैं। तो ये ड्रग डिजाइनिंग के अंदर भी हमें प्रोटीन स्ट्रक्चर की प्रिडिक्शन जो होती है वो हेल्प करती है। अब इसके आगे अगर हम बात करें कि उसकी जो कंप्यूटर सिमुलेशन बेस्ड ऑन एनर्जी कैलकुलेशन, इसके अंदर हमें कंप्यूटर की सिमुलेशन हमने एनर्जी बेस्ड करनी होती है, यानी कि जब हम फिजिकल और केमिकल प्रिंसिपल्स लेंगे कि फिजिकल और केमिकल वेरिएशंस क्या आ रही हैं, उसके अंदर बीच में रिलेशनशिप क्या आ रहा है, उस सबको लेते हुए हम इसकी प्रोटीन प्रिडिक्शन इसका स्ट्रक्चर प्रिडिक्ट कर रहे होते हैं। सेकेंडली हमारे पास थर्मोडायनेमिक इक्विलिब्रियम होती है जिसके अंदर हमारे पास मैक्सिमम फ्री एनर्जी होती है। थर्मोडायनेमिक इक्विलिब्रियम हमें बता देता है कि प्रोटीन की स्टेबिलिटी वगैरह कितनी है और उसके अंदर क्या सब कुछ होगा। थर्डली हमने बात की थी ग्लोबल मिनिमम फ्री एनर्जी ऑफ प्रोटीन सरफेस की, कि उसकी जो सरफेस है उसके अंदर मिनिमम फ्री एनर्जी कितनी है, उसकी जो एनर्जी की कैलकुलेशन वगैरह होती हैं वो हम इससे कर सकते हैं। फिर हमने बात की उसकी अप्रोचेस की, कि हम इनको प्रोटीन स्ट्रक्चर्स को अप्रोच किस तरह कर सकते हैं। उसमें हमारे पास 3 स्ट्रेटजीज आती हैं। सबसे पहले हमारे पास आती है होमोलॉजी बेस्ड। होमोलॉजी बेस्ड में अगर हमें एक प्रोटीन का स्ट्रक्चर पता है और हमें दूसरी का पता है तो हम ये चीज प्रिडिक्ट कर सकते हैं कि इन दोनों के अंदर सिमिलैरिटी कितनी है, इन दोनों के अंदर वेरिएशंस वगैरह कितनी आ रही है, इनका स्ट्रक्चर जो आ रहा है जो कि उसके कॉन्स्टिट्यूएंट्स हैं उसके अंदर उनका लिंक्स क्या है, अब उनका लिंक क्या आपस में, कि हम होमोलॉजी बेस्ड में करते हैं। फिर थ्रेड प्रोटीन सीक्वेंस हमारे पास आता है जिसके अंदर के हम उसके थ्रेड बेस्ड स्ट्रक्चर्स यानी कि उसके अंदर जो थ्रेडिंग प्रेजेंट होती है उसके थ्रू हम उसकी अप्रोच करते हैं प्रोटीन स्ट्रक्चर की। थर्डली हमारे पास आता है हायरार्किकल मेथड्स, यानी कि जो इंसेस्टर्स वगैरह से क्या चीज अडॉप्शन वगैरह चल रही है वो हम इसके थ्रू करते हैं प्रोटीन स्ट्रक्चर की।
Great. So the first of the method in a knowledge based method is homology modeling, and for homology modeling we need homologues of protein structure. So we need to have the homologues of protein structure. First of the thing we need, we can do backbone modeling, side chain modeling. So backbone can be modeled, side chain can be modeled separately as well. Fail in the absence of homology. So the homology modeling will fail if there is no homology. So because its basis is homology, it will need the similar sequence, and if similar sequence is not available, the homology modeling is not possible. And the basis of protein with similar sequences tend to fold in similar structure. Actually, this is the principle of this. So similar sequences will tend to fold in similar structure. So that will mean similar sequences will have similar structures. And has been observed that even proteins with 25% identity fold into similar structures. You already know how to determine the identities. So if two sequences have 25% sequence identities, only 25% sequence identities, the two proteins tend to fold into similar structure. That will mean we will call two sequences homologous. Homology are homologous if they are sharing 25, at least 25% sequence identity. For less than 25% sequence identity, don't go for homology modeling. If we have a sequence and that sequence shares like 80% identity with another sequence, that's well done, go for it, go for homology modeling. If 50% identity, that's okay, go for homology modeling. Even if 25% identity, go for homology modeling. But if that is less than 25% sequence identity, don't opt homology modeling. Am I clear?
Yes, sir.
Okay, and the next to it is it does not work for remote homologs. That is less than 25% pairwise sequence identity. The homology modeling is not recommended. And in the homology modeling, like what is given, a query sequence will be named as Q. That will be a query sequence. We will be having a query sequence. We will be given with that a database of known protein structures. We will be having a database like the PDB, protein structure database, and that database will already have the protein structure, and we will be having a query. So these two things we already have. What we need to find the protein P structure such that P has high sequence similarity to the Q. So we will find P structure and that will have high similarity with this sequence. Now in return, what will we get? P structure is an approximation to Q structure. We need to determine the Q structure. That will be an approximation of the P structure. And next to it is threading method. Threading method is a new way of fold recognition. So it will be for fold recognition. Threading method is also known as fold recognition method. So fold recognition is this method is and this sequence is tried to fit in known structure and motif recognition. It is used for motif recognition, loop and side chain modeling. It is not for the backbone modeling. And fail in the absence of known example. If we don't have known example, this threading method is will fail. So in this, the given is sequence of protein P with unknown structure, a database of known folds. So here the known fold database will be given. What we need to find most plausible fold for P, evaluate quality of such arrangements, and places the residue of unknown P along the backbone of known structure and determine the stability of side chain in that arrangement. So here normally folds are known, and through those folds the structures are determined. And in hierarchical method, this is the third of the method, that is are predicted instead of predicting the tertiary structure. So intermediate structures are predicted. The secondary structures are actually predicted from the amino acid sequence. So here the tertiary structures are not predicted. Here the intermediates are first predicted, and prediction of the backbone structure. The secondary structure is first predicted, beta turns prediction, and that's it. So tertiary structure prediction is then made. So first of all, intermediate structures are predicted, and then for the prediction of tertiary structures. Here the limitation is that the accuracy is only 70 to 85%. That is a limitation. So, and the next of the methods is ab initio methods. This is not a homology based method. We don't have any prior structure available, and its aim is ab initio mean starting from the zero, starting from the scratch. We don't have any structure known, and we want to determine the structure. So we have a method available, and that is ab initio method. And in this method, the aim is to find the fold of native protein by simulating the biological process of the protein folding. G. Mehak, you want to ask something? Okay, so the aim is to find the fold of native protein by simulating what? By simulating the protein, the biological process of protein folding. So here the biological processes are first determined, and then for the protein fold, and then to the structure. And it's a very difficult task because a protein chain can fold into millions of different conformations, and use it only when no detectable homologs are available. If no homolog is available, go for ab initio modeling. That will mean if there is less than 25% identities of the homologs available, so go for ab initio modeling. And method can be useful for fold recognition in cases of extremely low homology, that is convergent evolution. And example is iTASSER. So go on this site and just give a sequence to this site, and that will give you the protein structure, because it is based on ab initio methods. And the next is modeler.
सर
G.
सर ये जो बायोलॉजिकल प्रोसेस ऑफ प्रोटीन फोल्डिंग है इस पर अगर एक्सप्लेन कीजिए.
सो यहाँ पे इस तरह करते हैं कि ये देखते हैं कि कोई एक प्रोटीन हो और कोई और प्रोटीन हो जिसके फंक्शन सेम हो, या कोई एक like in a biological process, like if we have a protein that has same function to another protein, and these two proteins has same function. So now when I check the protein, it don't have any homology with another protein, but its biological process is same with another with another. So when I need to model this my own protein, this will be like this. It don't have any homology with other structures. So this method is available, and that is based on the biological functions. So the biological functions are then first determined, then the protein folding are then determined according to that biological functions, and then the protein structure is determined. But we don't need to like to do it ourselves. There are algorithms available, but those algorithms are basically based on this process, like they are based on the biological process of protein folding. So they will be having a database of the the proteins having biological processes, and through that it will determine the protein structure. Am I clear?
Yes sir.
सर मैंने एक बात पूछनी है.
जी जी.
आपने होमोलॉजी में बताया था कि हमारे पास दो प्रोटीन्स के सीक्वेंस हैं, एक का हमें स्ट्रक्चर पता है और दोनों के अंदर अगर मोर देन 25% वो होमोलोगस होंगे तो मींस कि दूसरे का स्ट्रक्चर और वो दूसरा भी सेम फंक्शन परफॉर्म कर रहा होगा, यही बताया था. और यहाँ पे हम लोग ये देख रहे हैं कि फंक्शन सेम है लेकिन उनके अंदर होमोलॉजी नहीं है, तो ये चीज कंफ्यूज कर रही है.
ओके ओके. वन बेसिक थिंग शायद हम ये कवर नहीं कर चुके. एक बेसिक difference is, I think we have covered that. which sequence will be, which of the three will be more conserved? Like if we have a sequence, if we have a structure, and if we have a function, these three things, if we have, so the sequence will be tending to have more mutations, that is less conserved if we compare with the structure. Structures are more conserved. That will mean even if we have different sequences, if we have differences in the sequence, they will have same structure. Like हम देख चुके हैं ना कि वोबल बेस में कोई सीक्वेंस चेंज आ जाए, लेकिन वो प्रोटीन जो है वो सेम बनेंगे, सेम स्ट्रक्चर बनेंगे. So if there are and even if there are changes in the protein sequence, there will be a difference in the protein structure. And next stage is, if there are differences in the protein structure, the function remain the same. ये देखा गया है कि even if there are different structure, they can give rise to the same function. So that will mean in the evolution, in the biological processes, function is more conserved than the structure, and structure is more conserved than the sequence. समझ आ रही है? So if we have less than 25% identities, so don't go for structure, for homology modeling. We still have to rely on the function, because function is more conserved than the structure. So even the structure is not conserved, but the function, the two proteins are performing the same function. So from that we can determine the structure, like that they will have a bit similar structure. And we will go for even the folding will be same. If the structure, not the structure, the folding will be the same. So based on that, go for ab initio modeling. Am I clear?
Yes, sir.
Okay, and next is a modeler, and modeler is actually the protein structure modeling, and this modeler is too much famous in the context of homology modeling. If you want to go for homology modeling, go for modeler. Modeler is the basis of actually different tools. The Swiss plot is the basis of it, and there are different techniques that do homology modeling, and they are based on modeler. And what actually modeler is a tool that is used for homology or comparative modeling for protein three-dimensional structure, to determine protein three-dimensional structure. And the user provides an alignment of a sequence to be modeled with known related structures. So we will provide an alignment of sequence to be modeled with known structure, and the modeler automatically calculates a model containing all non-hydrogen bonds. So it will calculate the structure that contains all the bonds and atoms except हाइड्रोजन. सो हाइड्रोजन नहीं होंगे, बाकी सारे जो बॉन्ड्स हैं, एलिमेंट्स हैं, वो उसमें मौजूद होंगे. And for detail, you can go on this site, just click on this site and it will go you through the modeler site. ठीक है, तो ये modeler का website है, go through it and you will understand how it works. So initially, modeler, what it do, align sequence with structure. It will align sequence with the structure. So this is the template structure, the red one, and the target sequence. The target we want, which we want to structure. We need to have the structure of this sequence. Like I have a gene like insulin gene, and I want to know that its structure. So what I will do, I will need a template structure, and my insulin gene sequence, I will take that, I will align that, and will get my gene
structured. And this is extract spatial restraints. It will extract the spatial restraint, the spatial distances, and after that it will satisfy the spatial restraint and give us the structure. So modeler is an automated approach to comparative protein structure modeling or homology modeling by satisfying spatial restraints. So spatial restraints को ये satisfy करके ये structure बनाएंगे. And input हम क्या देंगे? An alignment of sequence to be modeled. So sequence to be modeled, the sequence which we need to model is the target, and we align that sequence with related 3D structure, the template. So the template is the sequence, the structure according to which we want to model the sequence. And the output will be the 3D model for target sequence containing all main chain and side chain non-hydrogen atoms. So okay, so the output is a protein 3D model. We will get the 3D model of the protein sequence containing all non-hydrogen atoms. So after that, the important feature of the method is that the spatial restraints are obtained empirically from a database of protein structure. From a data, we will be having a database. From that database, we can get protein structures. Okay. So how it works, this is the flow through, the flow diagram, and that is the start, how we start. We will be having a target sequence. We will be having a template. First, align both. It will align our target. This is our target. We need to make it a structure according to this template. So align them, and after that alignment, it will be the target model, this model. So it is now model. And if you can see, these two models looks like the same. Maybe it will be having a little bit differences, and those differences will be the interplay of the sequence. And after that, we can get the diagram, the different parameters. According to those parameters, we will know how much the structure is reliable. We will be talking about these in detail in the coming slides. What are they? How the dope score and the soap score and these sort of scores. So these scores are actually they evaluate the structure, if that structure can be relied upon or not. So what it is like, identify related structure. First of all, we need to select the template. So identify related structure. So select templates from those structures. Align target sequence with the template. We need to align them. After that, build a model for the target using information from the template structure. So the model building is the next step. And after that, evaluate the model. Model evaluation is the next step. And if the model is okay, we need to evaluate through different procedures. After that, if that is okay, so that ends. But if that is the reliability is less, the evaluation score is less. So if that is less, so the model is not okay, go again from this. So we already aligned this, so go again from this point, or we need to select some other templates and start from here again. So this is how the protein modeling works. The protein model actually works. And for modeler, the necessary resources, we need a hardware, like we need a computer with Linux, or or we have a Macintosh system, or we have Windows, like the operating system is Windows, so XP or later, so they will work, the modeler will work on these systems. The software we need to download and install from this site. Just click on this and you will go to the site from where we can download the software. Modeler 9.13 are, if there are more recent version available recent days. Files we need to download. The complete protocol can be downloaded from this for Linux or for Windows. And we have an example, and that example is modeling lactate dehydrogenase from Trichomonas vaginalis, that is TVLDH. And this protein, this need to be modeled based on single template using modeler. So this example uses modeler, and this actually model this protein. So steps involved is searching structure related to this protein, searching the template, aligning the template with the sequence, and after that model building. And after we get a model, protein model, we need to evaluate that model, either that model is correct or not. And for visualizing how this works, we have a short video for that. अभी तक समझ आ गई है? Am I clear to all the class or not?
Yes, sir.
Yes, sir.
Okay, so सिबगा समझ आ गई आपको?
एक्सक्यूज मी सर
जी
सर, मॉडलर में हमारा फोकस नॉन हाइड्रोजन पे क्यों होता है? हाइड्रोजन को क्यों नहीं कंसिडर करते हैं हम?
हाइड्रोजन असल में वाटर मॉलिक्यूल्स भी आ जाते हैं. वाटर मॉलिक्यूल्स जो हैं वो फिर गेट्स प्रॉब्लम, एंड नॉर्मली इसमें हाइड्रोजन बॉन्ड्स को कंसिडर नहीं किया जाता. दैट इज नॉर्मल प्रोसीजर. इसमें लाइक आप अगर हम क्रिस्टलोग्राफिक स्ट्रक्चर भी देख लें तो उसमें भी हाइड्रोजन बॉन्ड्स जो हैं वो रिमूव किए जाते हैं. हाइड्रोजन बॉन्ड्स एक्चुअली सेकेंडरी स्ट्रक्चर प्रेडिक्शन में भी बहुत सारे हाइड्रोजन बॉन्ड्स इन्वॉल्व होते हैं. सो उन हाइड्रोजन बॉन्ड्स को देखा नहीं जाता, जस्ट कोवैलेंट बॉन्ड्स को देखा जाता है, नॉन हाइड्रोजन बॉन्ड्स को देखा जाता है. क्योंकि हाइड्रोजन बॉन्ड्स जो हैं वो उसको फिर वो कंसिडर नहीं किया जाता, उसको रिमूव किया जाता है. एंड एक्चुअली मेजॉरिटी केसेस में अगर कार्बन के साथ दूसरे एटम अटैच है और एक एटम नहीं है, सो दैट इज कंसिडर्ड दैट इज हाइड्रोजन. ठीक है
जी सर, थैंक यू.
Okay, so if we could ask from the people. रबिया जवाद, आपको समझ आ रही है?
Sir.
ठीक है. एंड ज़हीम समझ आ रही है आपको?
जी सर.
पाकीज़ा समझ आ रही है आपको? ओके, यू हैव माइक प्रॉब्लम. तूबा समझ आ रही है? तूबा?
जी सर.
ओके. सो इफ वी गो थ्रू द वीडियो, प्लीज इसको देखें एंड आई विल आस्क क्वेश्चन फ्रॉम दिस वीडियो. एक्चुअली दिस इज इम्पोर्टेंट.
ओके सर.
are done with half of the procedure. In a modeler, actually, what we need, we need a query sequence, and a query sequence will be the sequence for which we want to predict the structure. Like, I have sequenced an insulin gene and I want to predict the structure of that gene, like that is insulin. I want to predict the structure of this protein. Now what I will do, what I will need actually to use this modeler tool. So to use this, I will need the query sequence, means the sequence for which I need to predict the structure. And I will need the templates. The templates means I have to have few structures already available in the PDB. You already know the PDB, Protein Data Bank. In this data bank, there will be already resolved structures. The structures will be available there. So we already have the structures in the PDB, and using those structures, we will like predict the structure of our own query. Now, in this case, in this very example, we will be having 4 templates. They call this as T template sequence 1, template sequence 2, template sequence 3, and template sequence 4. They use these 4 templates. And what is the query sequence with them? They call that query sequence as query sequence 1. So this is the query sequence, and these 4 are the template sequences. So along with these templates and with these queries, we need to have 5 scripts. So we need to have scripts files as well. And they call these scripts as script 1, script 2, this is script 3, script 3, and this is script 4, and after that script 5. So all you need is a query sequence 1, 4 templates, 4 or 5 or 6, but at least you need to have 2 templates. But in this case, they use 4 templates and 5 scripts files. So we are done with this, that they like took a sequence for which they need to structure them, to predict the structure. They already have the 4 template sequence by searching this sequence in the BLAST tool while making the repository as PDB. You already know that. And while making scripts files, we are done with making 2 scripts files, and the third script file will be done today. So check that out. And okay, this one was the script 3, where which we have to start from. इन्होंने क्या किया कि मॉडेलर टूल यूज़ किया है, ठीक है। मॉडेलर टूल में इन्होंने सबसे पहले क्वेरी सीक्वेंस, दे हैव क्वेरी सीक्वेंस, और इस क्वेरी सीक्वेंस को इन्होंने क्या किया, हमें अब टेम्पलेट सीक्वेंसेस चाहिए। ये टेम्पलेट सीक्वेंसेस फाइंड आउट करने के लिए ये लोग गए हैं इस पे ब्लास्ट टूल यूज़ किया है, ठीक है। सो दे यूज ब्लास्ट टूल, और ब्लास्ट टूल में सिंपल ब्लास्ट टूल नहीं है, ब्लास्ट टूल में इन्होंने क्वेरी सीक्वेंस दिया है और वहाँ पे एक ऑप्शन को टिक किया है जहाँ पे आता है सर्च विद इन, किस में सर्च करें, पीडीबी में सर्च करें। आप जब वीडियो देखेंगे एक दफ़ा आपने इस चीज़ को देखना है कि इन्होंने ब्लास्ट टूल में पीडीबी को के ऑप्शन को टिक किया है। तो पीडीबी में क्या होना है, लाइक इस सीक्वेंस के अगेंस्ट सीक्वेंस नहीं आने, इस सीक्वेंस के अगेंस्ट स्ट्रक्चर आने हैं। तो वहाँ से इनके पास बहुत सारे टेम्पलेट्स आ गए। अब जब बहुत सारे टेम्पलेट्स आ गए, इन्होंने उसमें से 4 टेम्पलेट्स को सेलेक्ट किया है बेस्ड ऑन मैक्सिमम आइडेंटिटीज। सो वो 4 सीक्वेंसेस स्ट्रक्चर्स सेलेक्ट किए, उनको क्या किया, उनकी आईडीज ली हैं और उन आईडीज को पीडीबी साइट पे गए हैं, पीडीबी साइट पे जाके एक एक आईडी को सर्च किया है और उन स्ट्रक्चर्स को डाउनलोड किया है। तो इन चारों स्ट्रक्चर्स को पीडीबी साइट से डाउनलोड किया। सो नाउ दे हैव अ क्वेरी सीक्वेंस एंड द 4 टेम्पलेट सीक्वेंसेस, और इन टेम्पलेट सीक्वेंसेस को यही नाम रीनेम करके यही नाम दिए, क्योंकि हमने सॉफ्टवेयर में इसको रन करना है तो सॉफ्टवेयर ने इन्हीं नाम से रिकॉग्नाइज करना है अगर हमने इन्हीं नाम को यूज़ करना है, ठीक है। अदरवाइज हमने सेपरेट डिफरेंट नाम रखने हैं, उसके डाउनलोड किए हुए होने हैं और उस फाइल को उसी नाम से फिर रिकॉग्नाइज करवाना है सॉफ्टवेयर को। बट इट इज़ ईजी टू यूज़ इन दिस वे। ओके, आफ्टर दैट वी विल नीड दीज 5 स्क्रिप्ट्स। ये 5 स्क्रिप्ट्स हमें चाहिए। ये 5 स्क्रिप्ट्स ये बनेंगे किस तरह? वी हैव टू गो ऑन टू द मॉड्यूलर ट्यूटोरियल में जाके, ठीक है, वहाँ पे ये इन 5 स्क्रिप्ट्स के टेम्पलेट्स मौजूद हैं। मीन्स के वहाँ से जस्ट कॉपी करें और वहाँ पे इनके ये अगर वहाँ पे टेम्पलेट सीक्वेंसेस के नेम्स चाहिए तो ये ऐड करें, अगर क्वेरी सीक्वेंस का नेम चाहिए तो ये ऐड कर दें, ठीक है। तो इस तरह के 5 स्क्रिप्ट्स हमारे पास आ जाएंगे। नाउ वी हैव अ क्वेरी सीक्वेंस, 4 टेम्पलेट सीक्वेंसेस एंड 5 स्क्रिप्ट सीक्वेंसेस। ये सारे सीक्वेंसेस हमने एक फोल्डर में डालने हैं, ऐड करने हैं, एंड we have to save them in the C drive। इसको C drive में हमने सेव कर देना है, ठीक है जी। फातिमा रमजान,
स्क्रिप्ट्स क्यों यूज कर रहे हैं?
स्क्रिप्ट्स इस वजह से यूज कर रहे हैं कि मॉड्यूलर टूल इन स्क्रिप्ट्स के लैंग्वेज को समझेगा, क्योंकि मॉड्यूलर टूल जो है इसको ये नहीं पता चलना कि हम चाह क्या रहे हैं, what we need actually। So the modeler tool will read the language, ठीक है। ये पाइथन स्क्रिप्ट्स हैं, पाइथन लैंग्वेज को ये मॉड्यूलर टूल समझेगा और उसको रन करेगा। हम आगे जाके यही डिस्कस करेंगे कि इन स्क्रिप्ट्स में बेसिकली है क्या, लेकिन फिलहाल I want to let you know what actually the procedure is, ठीक है। So scripts files ready, now we have to run modeler को रन करना है, modeler को ओपन करना है। modeler में फिर हमने C को लोकेट करना है और फिर C में ये फोल्डर जो है ये लोकेट होने वाला है, ठीक है। आफ्टर लोकेटिंग दिस फोल्डर विच कंटेन्स ऑल दीज फाइल्स, हमने फिर इसको रन करना है। सबसे पहले स्क्रिप्ट 1 को रन करना है। स्क्रिप्ट 1 में जो लिखा हुआ है मॉड्यूलर टूल उसको समझेगा और उसको रन करेगा, ठीक है। अच्छा, फिर स्क्रिप्ट 2 और ये स्क्रिप्ट 1 जैसे ही रन होगा, इफ दैट इज सक्सेसफुल वी विल गेट अ लॉग फाइल। सो जिस फोल्डर में ये फाइल सेव है तो वहाँ पे script 1.python है py, उसके साथ script 1.log उसका लॉग, उसकी लॉग फाइल भी हमारे पास, वी विल गेट दैट फाइल, ठीक है। अच्छा, फिर वी विल रन script 2 and ultimately we will get script 2 log file, and same for the other script file. After running the fifth file, the fifth script, हमारे पास उस फोल्डर में 5 different structures आ जाने हैं for this query against this query sequences, क्योंकि हम ये scripts फाइल बेसिकली हम ये देखेंगे अभी आगे जाके, उसमें हमने इनको कहा हुआ है कि उसमें लिखा हुआ है कि 1 to 5 मॉडल स्ट्रक्चर्स हमें दें, तो 5 मॉडल स्ट्रक्चर वो हमें दे देगा। हमने उन 5 मॉडल स्ट्रक्चर में सेलेक्ट करना है कि which one is the best structure to go forward with that। हमने वो स्ट्रक्चर सेलेक्ट करके उसके लिए हमें एक और विजुअलाइजर चाहिए होगा जिसमें हम प्रोटीन स्ट्रक्चर को विजुअलाइज कर लें। लाइक इन्होंने रेस विन टूल यूज किया है, रेस विन, ठीक है। हम काइमेरा को भी यूज कर सकते हैं, ठीक है। और भी बहुत सारे टूल्स हैं, अगर हमने इन स्ट्रक्चर्स को विजुअलाइज करना है सो वी कैन विजुअलाइज विद दीज टूल्स। तो दिस इज द होल थीम कि हमने एक्चुअली करना क्या है, ठीक है। समझ आ गई कि नहीं आई? इज इट क्लियर? ओके, दैट्स गुड, ठीक है। एंड so I will share these slides. इस स्लाइड में इस वीडियो का लिंक भी मौजूद है, you can go through this video, ठीक है। I will upload this today inshallah. Okay, so सबसे पहले हमने क्या किया कि searching structure related to TVLDH. So previously we have talked about, we will be having an example of this TVLDH and based on a single template. So we already have a single template as well. So the steps involved will be searching structure related to this sequence. Now selecting template, align this sequence with the template, then model building step, and then evaluation of the model. So conversion of the sequence to PIR file format. Now we need to understand what is PIR format. PIR format है क्या? So modeler क्या करेगा? This will use the PIR format to read, write sequences and alignments. So for modeler, PIR format is important and it could understand only PIR format. So the first line of the PIR formatted sequence consists of this thing. Like we will be having a greater the sign of the greater than, and after that we will be having like the P1 and semicolon and the name of the sequence. After that the sequence colon and the name, and after that 1 2 3 4 5 6 7. So these are 7 colons and 0.0 and then colon and then 0.00, and this is actually, this header is important for the modeler to understand. This will understand that this is the name of the sequence and this is actually the name, and after at the end of the sequence there must be steric sign. The steric sign will mean this sequence ends here and this is an amino acid sequence. And how you will name it? You will name it as tvldh.ali. So ALI format is actually this format, and so we will name it as tvldh.ali. So ALI is the extension. This is just like .docs, .pdf, like ppt x, and this is .ali. So this is important. You need to understand that. So sequence file in the PIR format is this now. So am I clear with that? And the uppercase amino acid letters will be used. So these are amino acid letters, the first single letter amino acid representation, and they will be in the uppercase. So next, PIR की समझ आ गई है class?
Yes, sir. Yes, sir.
Okay,
Yes, sir.
and okay, and next to it is searching for suitable template structure. So a search for potentially related sequences of known structure can be performed using, so this command, this name will be used, and in the modeler command, the command task is input the target sequence in the database of known structure. So known structures will be, so this file is important. We will also need this file, pdb_95.pir. So this PIR format is important. So this file is important to run the modeler. So along with a template, along with a query sequence, along with 5 or 4 templates, and along with the scripts files, we also will need this file, pdb_95.pir. So this .pir file is important, and this file you will get from the modeler website. You will also get this file, and returns a set of statistically significant alignments. You will get alignments of the query with the template structures. So now execute the scripts file using the command. So how you will execute that? So in modeler, site, you will execute in the modeler window. You will execute mode 9.13 build_profile.py. So this name, we have already named this as the query sequence one, script1.py. So we will add there mode 9.13 or whatever the version of the modeler you are using, and after this we will run script1.py. And at the end of the execution, a log file will be created with the name of script1.log. We actually name this as script1. So after that, you will get this log file. In the log file, the important things are, like, the most important column in the log file is the output, are the second, this second one, the second, 10th and 11th and 12th columns. So they are important. So this is the second one, this is the second column, the 10th column, this one, the 10th column, 11th column, and 12th column. These are important. And why they are important? The second column actually reports the code of the PDB that was aligned to the target. So all these are aligned to the target actually. So TVLDH code for these are the PDB codes. And the 11th and 11th column reports the percent sequence identities. So here are the percent sequence identities. The 11th, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 11. So these are the percent identities. So you can see this is 0. This is 28% identical. So this is 42% identical. This is 45. So you can see the best identities are with the 1 BDMA. This one, 45. So this has best identities. No, you can see, you can observe in this line. So this is the maximum identities shown here with the 1 BDMA. Actually, A is the chain. In the last, this A represent the chain A, B, and so on. So these are the chains actually in the last one. So, and the PDB sequence normalized by the length of the alignment indicated in the 10th column. In general, a sequence identity above 25%. So the sequence identity above 25% indicates a potential template. So unless the alignment is too short, that is. So two things we will need. We will be having, we will require more than 25% identities, and we will need alignment. We will need more than 100 residues in our sequence. And after that, A better measure of significance of the alignment is given in the 12th column with the E value. So E value is important. This is the expected value. So lower the E value, the better. So the 0 is the better value than others. So 0 means we can rely upon the sequence. So the lower the value, the better the alignment is. And 6 PDB sequences shown very significant alignment and identities, similarities to the query sequence with E value equals to 0. So you can see 0 expect value is this one, 1, 2, 3, 4, and 5 and 6. 1, 2, 3, 4, 5, 6. So these are the 6 values, but this is 7 here with the E value equals to 0. So anyways, so here an expected all the hits correspond to malate dehydrogenase. This was the query sequence, and they corresponds to 1 BDMA. This one 1 BDMA. This is 0. Actually, this TVLDH is the query sequence, so we will not consider this one. The rest of they are the template sequences. This is actually the query. The first one is the query one, and they are the templates. So we will be having in this case we got 6 zeros with expect values 0, 1, 2, 3, 4, 5, and 6. So they are 1 BDMA, 5 MDHA. This one 5 MDHA. This one 5 MDHA and 1 BP8A. This one and 1 CIVA. This one and 7 MDHA. This one and 1 SMKA. This one. So all these 6 will be selected by the modeler in the next coming log files. So now the next the next script file will be like in this case they use compare.py, but we rename that as script2.py. So it will align and compare the structures, actually like align dot compare structure. So this will, like you can see here, this is the language that has been used. This is how it starts and align. So we give the command like align the sequence, and for PDB, so the first one is the PDB structure, and again the comma, and the second one will represent the chain. So this is the structure, and this will be the chain of that structure. So we will be like the structure name is 1B8P, and the chain is A, and same is the case with other. So they used 1B8PA, 1 BDMA, 1 CIVA, and these are the same 6 sequences that has been selected here. So they are here, and after that model and the chain, and first the name and then the chain, and then the rest of the commands are to align 3D structure and so on. And the next is this excerpts from the log file compare are shown in figure 5.6. So this is how it's shown in the next, in this file it has been shown. But before that, this initiate with the environment, ENVIRON. So environment for this modeling run by creating a new version object called ENV here. So it introduces a new like object. So object is a different thing in the language. We will be discussing this thing in detail in our Python and R class.
So almost all modular scripts require this step as the new object is needed to build more other useful objects. So this will be the log file, and how this log file looks like. So check this out. These are actually those 6 template files, the 6 structures, and these are compared actually. And this is actually the phylogenetic analysis, phylogenetic tree dendrogram that has been used. So this shows that how to read this tree. And from this tree, we will select one of the template, one of the sequence. We will select one of the sequence against which we will model our query. So the objective of this step is to select the most appropriate single template structure from all possible templates. We need to select a single from all these based on this tree. So how we will select? The dendrogram shows that 1 CIVA and 7 MDHA are almost identical. So you can see these two are almost identical. And these are the values for resolution. So this is the crystallographic resolution. This will be given in the PDB file or on the PDB site. So first of all, we need to know these two are closer to one another, revealed by this dendrogram. So however, 7-MDHA, this 7-MDHA has better crystallographic resolution. So the lower the resolution, the lower the value of this resolution, the more the resolution is. So 2.4 angstrom versus 2.8 angstrom. So 2.4 angstrom is a better resolution and will be selected. So 7-MDH, we will select that. From the second group, so in this group, we will select this one. From the second group of similar structure, 5-MDH, this one, 1 BDMA and 1 B8P. So these three, we are talking about these three, has best resolution 1.8 angstrom. So in these three, 1.8 resolution is the best one. So 1 BDMA is the best out of these three. And out of these two, 7-MDHA is better. And so the rest is only we are remaining with only one structure, and that is 1 SMKA. And this is most structurally divergent among the possible templates. So this is, you can see this is most divergent sequence. This is farther from other sequences, other structures. But it is also the one with the lowest sequence identity. So with the lowest sequence identity, now you can see sequence identities file, this one. So this is 34. So out of all these zeros, means this is 34, this is 44, 44 is better, here 35 is lower, this is 45 is best even, and 42. So we need to check into this as well. We need more identities, more similarities, and the highest similarities will be selected. And after that, we will also check the resolution. So here we will also check the resolution along with the identities, and some are within the groups. So this 1 SMK is more structurally divergent, even though, but it has lowest sequence identity. It has 34% identities to the target sequence. So we will discard this one because it has lower identities. So now we are left with this 7-MDHA and 1 BDMA. So finally, in these two, which one we will select? This 1 BDMA will be selected because of the higher
overall sequence identities. We will check this, like it has overall highest sequence identities, 1 BDMA. It has 45% identities. So it has 45% identities. So therefore, we will select this one. It has even better resolution than this one. So therefore, we will select this one. And next is the TVLDS sequence in the this file. In the initial file, the script one file is to be aligned with 1 BDMA. So structure in the PDB file 1BDM.pdb. So here the commands are, so here we will align the sequence 1BDM model segment, and you can see we are telling the software to align tvldh.ali with what, with the with 1 BDMA. So you can see this one, tvldh. This is our query sequence, and we are assigning the modeler to align this tvldh with 1 BDMA, and this 1 BDMA has been selected from this one. So these two formats, .ali, .pap format, .pir format, we will be talking about this PAP format as well. So we have talked about .ali format. In the last, it will have a static sign, and it will have a greater than sign in the initial and the name and so on. And we already have talked about .pir format. PIR format was different. Now we will be talking about PAP format as well. So in the first line of the script, an empty object.aln, this one ALN has been introduced, and a new model object MDL, this one MDL, this one, into which the chain A of the 1B MD structure is read are created. So this 1BDM is read and created. This has been created, and after that it has been assigned to the modeler to align the two. And append also, this is the append model transferred the PDB structure to this model to align. So this will align the two structures, and the TVLDH sequence from the file .ali is then added to align using append. The align 2D, here you can see there will be align 2D, yeah, this one. So align 2D command, this command aligns the two sequences. So this will align the two sequences, and the alignment is written out in two formats. So it will be written in two formats. So in the output, what will be the output? See, the output will be in the two formats, .pap format and PIR format. So in these two formats, it will give the result. So write, so give us the result. So align that right. So write it in two formats, PIR format and PAP format. So it will give the output in two formats actually. And after that, after getting the log file of this script, the PIR format, now the PIR format is used by a modeler in the subsequent model building stage. So now we are aligning the TVLDH with the template. The PAP alignment format is easier to inspect visually. So PIR format, so these two formats, the output will be these two formats. So why we use PIR format? Because the modeler can understand this format. And why PAP format? PAP format is for us. We can understand PAP format easily. So you can see this one. So this one is the template, and this one is the query. And these two sequences has been aligned, and the statics shows conserved. We already have discussed how the conservation has been represented in alignment. And this is the alignment between sequences TV, LDH and one BDMA in the modular PAP. So this is the PAP format, and you can understand this alignment actually. So this is for our visual inspection. We can understand this alignment. And the PAP format, all identical position are marked with asterisk. You know this better than me. Once a target template alignment is constructed, modeler calculates a 3D model of the target completely autonomically using auto model class. So now this script is important. Looking at this script, you need to understand we ask the modeler to auto model TV LDH and one BDMA alignment .ali, and what the modeler know is we will be calling that as, we will write that as knowns. Modeler also already know, like we will give the sequences of one BDMA and the sequence TV LDH. So this is the query and this is the template. And we will be asking the modeler to assess the alignment and the model through 3 techniques, like assess.dop. We will be needing dop score, SOAP score, and dop score and the GA score. So dop score and GA score will be assessed by the modeler, and this hashtag, this will mean this is for us to understand. This is not for the modeler. So modeler will give us 2 values, this dop score and GA score. Okay, and we are calling the modeler to start from the model 1. So to execute model 1, like start from the model 1 and end model, how much models we need? We need at least 5 models. And after that, the command is make. And this will end here. This is how the language is. The modeler will understand what we need actually. So at the end, this will give us 5 probable models. And in those 5 models, we will assess those models with dop score and with GA score. So 5 models will be generated. An auto model object is created and called A. So this has been created and we call A. And the parameters are set to guide the model building procedure. Align file, name the file that contains the target template in the PIR format. We already have discussed this thing, and knowns are the structures that already we know, and we have saved this with these names, and the sequence defines the code of the target sequence. So the code of the target sequence. And okay, so the most important output file will be in the model single. Okay, so in this way, we report warnings, errors, and other useful information, including the input restraints and so on. And it will give us 5 structures, 1 to 5, pdb.pdb, which contains coordinates of the 5 produced models in the PDB format. And the models can be viewed. You can view this in the Chimera, in the race mode. You download Chimera, a race mode software, and you can visualize these 5 models in that. So in this, you can see if several models are calculated for the same target. If we need to calculate several models, the best model can be selected by picking the model with the lowest modeler objectives function. So based on the modeler objective function, the dop score and SOAP score are reported at the end of the log file. So when we run this file, we will get a log file, and in that log file, we will select the structure, the best possible structure out of the 5. So in this example, the second model, this has the lowest objective function and is selected. So if you can see this one, this is assess, and okay, the dop score, give the dop score, and so here it's not given. So once a final and that was shown in the video actually. Once a final model is selected, there are many ways to further assess it in this example, dope potential in the modeler is used to evaluate the fold of selected model. So we can evaluate the fold of the models as well. So here now which structure is being selected? TVLDH dot this second number model is being selected based on dope score, GA score, and the other value like the objective function. And the script evaluate the model of the dope potential and energy profile. So this is important. Class, please try to understand this. The energy profile is additionally requested, smoothened over 15 residues window, normalized by the number of restraint. The profile is written, which it can be used as input in the graphing GNU plot. In the, for instance, the GNU plot, this is a graphing program. And you can see this graph. This graph is important. Please try to understand this. This the comparison of pseudo energy profiles of the model. So the red one is our model. So this is a graph for our model, the red one. And the green one is the template. Means the template that has been taken from the PDB. And now we have the model in the red one. So we can see if the red one aligns green. So अगर ये दोनों lines जो हैं वो अगर overlapping हो, so it will be better. लेकिन और ये line जो है ये threshold है, ठीक है, this one dotted line. So अब यहाँ पे देखें इस area में ये green और red align सही नहीं हो रहे, difference ज़्यादा है इन दोनों के दरमियान. So अब हम क्या कहेंगे कि अगर ये मॉडल हमारे पास आ गया रेड वन, फिर हमने देखना है कि रेजिड्यू नंबर 100 से 98 या 95 से लेकर 120 तक ये रीजन जो है ये इंपॉर्टेंट तो नहीं है. अगर ये इंपॉर्टेंट रीजन है मींस दिस इज द डॉकिंग साइट या एक्टिव साइट है, तो एक्टिव साइट में ये सही मॉडल नहीं हुआ, ये इस मॉडल के अकॉर्डिंग नहीं है. So this will not be a good model then. So हमने जो मॉडल लिया हुआ है we will reject that based on the differences. So अगर ये important जगह नहीं है और कोई important जगह ये वाली है या ये है, so this aligns better. ये देखें ये एक दूसरे को overlap कर रहे हैं, so this means ये area सबसे best है alignment में, ठीक है. बाकी ये region is different. These regions are different. So हमारा मॉडल जो है वो इसको ओबे नहीं कर रहा टेम्पलेट को इन रीजन्स में, ठीक है. समझ आ रही क्लास?
Yes, sir.
So after that the profile can be calculated. अब ये इसकी explanation है. The profile can be calculated for the template structure and a comparison between the true profiles. ये comparison हमें देगा कि true profiles जो हैं वो उसको compare किया जाता है. The dope score profile shows clear differences between the two profiles for the long active site between the residue 90. अब यहाँ पे हम देखें अगर हम residue number 90 और 100 residue देख लें, so अगर profiles for the long active, so अगर ये active loop है between 90 and 100 and the long helices at C terminus end of the target sequence, अगर यहाँ पे ये C terminus है ना, ये N terminus है and this is the C terminus, तो यहाँ पे differences बहुत ज़्यादा हैं C terminus पे, ठीक है. So target और जो template sequences हैं इनमें differences बहुत ज़्यादा हैं यहाँ पे और यहाँ पे, ठीक है. So the latter region is well resolved in both the template and the target structure. However, probably due to unfavorable non-bonded interactions between, यहाँ पे जो non-bonded interactions हैं, it is reported to be high energy by the dope. So इसकी energy बहुत ज़्यादा है यहाँ पे और इसके बाद ठीक है. And the same active loop has a better profile in the template. So active loop का profile जो है वो यहाँ पे अच्छा है, ठीक है. So और डोप स्कोर जो है ये पहले हम डिस्कस कर चुके हैं, the lower the better. तो डोप स्कोर जितना कम होगा उतना बेटर है. तो यहाँ पे अगर देखें तो हमारे जो ग्रीन वो है this is the template. So इसकी ओवरऑल एनर्जी जो है ये डोप स्कोर जो है इसका कम है,
ठीक है। तो यहाँ पे डॉप स्कोर ये ज्यादा आ रहा है यहाँ पे, ठीक है। सो ये दिस मॉडल इज नॉट अ गुड मॉडल, which strengthens the argument that the model is probably incorrect in the active site region. So active site region में ये मॉडल जो है वो अच्छा मॉडल नहीं है. And these are some of the databases you can search upon, and these are protein sequence databases. You can go through it. Domain and super families के कुछ sites हैं, कुछ databases हैं. Protein structures and models. These are the some databases for protein structures and models. Web servers हैं कुछ, ठीक है. And these are some miscellaneous sites. Sequence and structure based alignments के लिए कुछ tools हैं, cluster W and muster muscle and so on. And these are the structure databases and web servers. And these are further some more websites, like you can go for alignment modules in molecular graphics programs. So if you need to have like visualize some protein model, so go for Discovery Studio. It's the best visualizing tool. PyMol, Swiss PDB. So this is a viewer, actually structure viewer, UC SF Chimera, and so on. So these are the web servers for comparative modeling, threading and refinements. So these are the tools you can go, and this will automatically give you the structures. WebMod, File2, Robetta, Swiss model. So अगर आपने खुद मॉडल नहीं करना, so automatic tools हैं, यहाँ पे सिर्फ सीक्वेंस दे दें और ये इन्होंने आपको मॉडल बना के दे देना है. So these are the web servers for this, for those. So these are some further programs like Modeller. So we have covered this Modeller and the rest of as an example. And these are certain other tools, Rosetta and more. So these are some evaluation tools to evaluate how to evaluate protein structure. So through Rosetta, through this SCWRL, we can evaluate. So we can evaluate the quality of our structures. So these are array score, model evaluation, Pro Q2, Pro check, and Q means Swiss model, verify 3D, what check. So these are different tools we can evaluate our structure with. And these are the methods, methods evaluation. So these are the some tools for methods evaluation as well. Go through these and you will understand easily what are they. At least you have